today. So the first part that we'll be discussing today is the research designed to optimize the quality of any clinical trial or research that we are going to conduct. There are many types of research that we can design, and this will mostly depend upon our research project aim, the hypothesis, and the problem that we want to investigate. So the first part is the qualitative research. In qualitative research, this is the basic or the introductory type of any studies that we want to gain or obtain information regarding a particular problem that we want to look after that into conducting clinical trials. So qualitative research will focus on the quality, therefore will not be regarded with recruiting a high participant number, but rather on the quality of the information that is to be collected. So why do the patient and public get involved in research, in particular during the qualitative research? This is due to the ethical right, the democratic right, the utilitarian argument, and mandatory for some health research institutions, and to also avoid any waste research. So including patient and public in the qualitative research type will help us to gain more insight into the particular problem that we are investigating. So in the research cycle, at each step, we can include the public, which is known as public involvement. For example, in identifying and prioritizing the issue that we want to investigate, we can include the public. They can identify any relevant research topic through consultation, for example. Also, in the designing of the study, they will ensure that protocol, for example, are appropriate. They will assist with recruitment and retention strategies. They can define the outcome measure and assist in every step within the research cycle. Also, they can help in disseminating of the results by advising on venues or jointly presenting the research or contribution to its publication. So, what is qualitative research? The first thing that we need to understand, it has more significant value of the question that we seek to answer about a phenomenon, not regarding how much participant number, but rather on the quality of the information that we are collecting. So there are different ways of knowing or understanding a problem. These are philosophical methods, including the positivism, which is a stable reality, observable, and value-free that is aligned to the natural sciences. There is also the interpretivism, which is not a search for the reality of the world, but also on how people interpret the world or how they view it in their own view. So understanding what the assumptions are made at the planning stage of the research will help us in the design process. So the difference between qualitative and quantitative is that there is always interpretation to the data in the qualitative type. So it is not like there is an absolute answer to every set of data. For example, when we get bands or on a gel, for example, or we get some kind of numbers, it has to do with how you set up the machine, take the background, and how we control the external variables, not on rather interpreting the large occurrence in numbers of participants. So qualitative research is a set of interpretive material, practices that make the world visible, and these practices transform the world. So they turn the world into a series of representations, including field notes, interviews, conversations, photographs, or recording of memos in itself. So qualitative research study the things in their natural setting, attempting to make sense of or interpret the phenomena in terms of the meaning that people bring into this. So the key features of the qualitative approach in the research question, they focus on how and why rather on how many regarding the number. And for the data collection, these include close contact between researcher and research participant with flexibility and responsiveness to the context and to the accounts. And regarding the data that is yielded, these include rich, detailed, and extensive information in the content, but have a fewer cases than the quantitative type of research. Also, for the analysis, it will allow for emergent concept and involve the description, the classification, typology, and explanations with findings that emerge from the data in itself rather than a previously categorized idea. And regarding the output, it will have interpreting social meaning and representing the social world of the participants. So when should we use qualitative research? For example, in an HPV vaccination study, we need quantitative study to estimate the size of the target population, the scale of vaccine production, the service framework for providing the maximum coverage, for example. And we also need qualitative study to find out what parents, adolescents thinks about the vaccine, why do they believe what they do, and how to overcome these challenges. So an example of this is the VOICE trial, which is the vaginal and oral interventions to control epidemics. In a multi-set HIV pre-exposure prophylaxis trial, the target was young women in South Africa, Zimbabwe, and Uganda. They were given a daily application of 1% of tenofovir vaginal gel plus a daily oral intake and enteric bond. So the results of the trial found no effectiveness for the three products tested among 5,000 women at 15 different sites. So the widespread product, non-use, including by assessing the plasma level, despite the high end of the study retention rate and the high self-reported product use. So to explore further into the issue with this intervention, they conducted a qualitative exploratory study in which it was implemented between July 2010 and 2012 August. So 102 participants only were recruited in the South Africa region, and the focus group discussing in-depth interview at the study with clinic with a series of ethnographic interviews found that the reason for non-use were HIV stigma, the fear of being labeled as having HIV, ambivalence about the research process, confusion about the use of the antiretroviral to prevent the infection, and the need to balance the trial participation with other priorities and social relationships. So when we have conducted a qualitative study, as in the previous example, we understood what the problem was in terms of the participant perspective and how to overcome these issues in the future. So the different methods and different purposes include in the quantitative research, it focuses on numbers, while the qualitative research focuses on words. Also quantitative test the hypothesis, while qualitative will produce theories and hypotheses to examine later in a clinical trial. So in the quantitative, it have closed question with detached researcher, but in the quantitative, it have open-ended questions and more involvement on the researcher part. So the qualitative research focus more on the researcher involvement with the participants to understand their point of view. So regarding the context, in the quantitative research, the extent of the existence forms of homelessness, for example, will include characteristic of homelessness of people, while the qualitative will study the nature of different forms and the meaning of being homeless, for example. So to ensure the optimum study design, we need to consider the sampling method in which we can use. In the qualitative sampling, this will include in-depth exploration with data and context, while in the quantitative, this will measure the external extent with the statistical correlations. So the sample recruitment in the qualitative study will map the diversity, including key groups and dimensions, with a smaller sample size, while the qualitative type will have statistical representation with the exact mirror of population and the large sample size with robustness of the estimations. Also, the qualitative type will have a sample that is purposive, in which it will be a deliberate selection on the basis of knowing their characteristic, while the quantitative will be a random probability sampling technique. So there are 4 sampling techniques that we can use. These are the purposive sampling, the theoretical, the convenience, and the snowballing method. In the purposive or the criteria-based sampling method, the sample is selected with a purpose to represent a type in relation to key criteria that is suspected to be relevant. For example, based on some socio-demographic or experience behavioral role. In this type, it will focus on 2 aims, ensuring that all relevant key criteria are represented. It will also include diversity within each criteria. Different approaches to this sampling method is the homogeneous, the heterogeneous, the extreme cases, and the key informations. While in the theoretical sampling method, the samples are based on their potential contribution to the development and to the testing of the theoretical construct of the study. So association with the development of the grounded theory will include initial sample being selected, data being analyzed, and further samples selected to refine the emerging categories and theories to be assessed. So this is useful in exploratory research where little is known about a particular phenomena in general. While in the snowballing method, these include series of introductions to a new sample from a research participant. This will be useful for hard to reach, easy to ignore groups in which they can be included. So this will also be the convenience sampling in which we choose a sample according to ease of access, but it lacks a clear sampling strategy. So some of the methods for inquiry in qualitative research include observations, in which they may be combined with participation. Also, archives or other documentary sources, including the media. And it will also include interviews such as semi-structured, narrative, or one-to-one interviews, or even focus groups. So interview types can be structured interviews that is used in quantitative surveys as well. This will set standard questions or standardized question methods. It will also have a fixed wording and order of questioning. It will also have predetermined categories and will be part of a larger sample survey. So in the semi-structured interviews, these are the qualitative type of interviews that will be more flexible and will have standardized interview schedule, including topic guide, or it can also be exploration of emerging themes and ideas with limited reception or disclosure on part of the researcher. There is also the unstructured interviews. These are the qualitative type and are interview style unstructured with some gentle guidance only and biographical stories or narrative to better understand the issue that is to be discussed. So this is an example of a topic guide. This will be a flexible guide with document scopes and coverage of the interviews, as well as key sections with key subtopics and ordering of the topics and beginning with easier, less sensitive topic and moving on to a more sensitive set of topics. So these can include pilot interviews and producing a topic guide that will help better guide the process. So the question for breadth and depth will include short open question versus the close. This will avoid leading the questions or doubling the question again, which will lead the participant to change their answer, for example. Also, we should have content mapping to raise issues that are prompt to the issue, and the content mining to explore issues that are emerging. That will include elaboration, exploration, explanation, and clarification. So when we are conducting interviews, we should build rapport and trust with the participant. Also, we should have neutral and reassuring approach, avoiding self-disclosure and allowing the respondent time to think. We should also listen and role as researchers, not as a counselor or advisor or a healthcare professional. We should also audio record and match the interviewer with the interviewees in order to ensure that we have the best outcome from the study. There is also the focus group type of interviews. These can include 6 to 8 participants. They will have interactions between the participants and themselves, and it will have composition that is carefully selected to encourage exchange of views and experience in general. So in the in-depth interview versus the focus group, the in-depth interviews include personal stories, detailed or complex issues, and sensitive issues that the participants feel free to discuss. Where in the focus group, these are generated by the group interaction and will have broader understanding and not too sensitive information as it is shared within somewhat of a large group. So in the qualitative data analysis, the aim of our analysis is to understand the phenomena that is being studied, also to go beyond reporting, but rather to interpretation and to identify the themes and the sub-themes that are emerging. And we should be reflexive in terms of our analysis to ensure that we produce the best outcome of the data that is to be analyzed. So when we are managing the qualitative data, the data security is important as the qualitative data may include personal opinions or details that are sensitive. We should have coded ID used to sort out the data and the transcript in general, and it should be protected by confidentiality and anonymity of the information when we are reporting these results. And the analysis in this part of data begins early on as soon as we have developed a topic guide and conducted our first interview. So this is a iterative process with new insights from the initial data collection and subsequently incorporated into topics guide and into sampling methods. So the usefulness of taking notes during the data collection process during the field diary is of paramount importance in this process. So the framework analysis process includes data familiarization by reading the interview transcript, listening to audio recording, and using field notes in general. So creating the thematic framework, developing a coding scheme of themes, and indexing or coding, signposting the interesting byte, not the meaning in itself. Also charting by creating a thematic chart and providing a picture of the data. Also allowing for visual assessment across the individual cases and themes, and mapping and interpretation by exploring the associations between the themes and developing some sort of conceptual understanding and categories to capture the essential meaning that is going beyond the descriptive data, but rather into interpreting the hidden meaning behind their response. So the framework analysis approach allows within and between cases analysis. It will also increase the transparency of the data analysis process, allows the existing theories and ideas about the area under study to be taken into account within our analysis. And also the analysis is both inductive and deductive, with some themes that are prioritized, but some are emergent from the data, plus the new analytical themes that develop during the process. Other forms of data analysis include the grounded theory analysis, in which it will generate analytical categories with identifications of relationships between them, as well as the data collection and conceptualization that will continue until the categories and the relationships are saturated and no new data are available to be added. Where in the content analysis, this will focus on counting the numbers of times that something is mentioned in the field notes or the transcript as though as a rough estimation of the frequency. So in the hermeneutical analysis, we are not looking for the objective meaning of the text, but rather the meaning of the text for people in a certain situation that is shared on the construction of the meaning in general. Where in the discourse analysis, this is a linguistic analysis of the ongoing flow of communication. It will find pattern of questions which dominates time and how the other pattern of interaction are being viewed in general. So in the phenomenology, this is how the individuals experience the world, and it will emphasize on the idiosyncrasy meaning to the individuals and not sharing the constructions as such as with the hermeneutical analysis. There is also the narrative analysis in which this will explore the participant stories and what they choose to share about themselves. So The main concerns in qualitative research are the quality, the validity of the information, and the generalizability of the information in terms that we can uh, spread out the information to the rest of the population. So in the quality of the qualitative research, it is a part of a much larger epidemiological debate about the nature of knowledge that is produced by qualitative research. So how can we judge the quality of qualitative research? There are, of course, some standards that we can follow or guidelines, including the CORIC, which is the consolidated criteria for reporting qualitative research. This is a 32-item checklist that was developed in 2007 to interpret or to assess the validity of the criteria of qualitative research. There is also the RATS, which is the relevance, appropriance, and transparency with the soundness of the interpretation of the research process. And regarding the validity of the research, these are the transparent and comprehensiveness of the research, also to provide the context for the research and give clear evidence for interpretation, including analysis of deviant cases, as well as the triangulation to provide the overall interpretation, including the multiple methods that is used or the data sources, or even assessing previous research to ensure that we have the best outcome in these methods. So all of these should be reflexive in terms of that the researcher role in affecting the research process, the outcome and the interpretation, and how the data are collected, the intellectual personal bias, the priori assumptions, and the personal characteristic of these data. So regarding the generalizability, these include three kinds, which are the representational, whether what is found in a research sample can be generalized or to be held equal to the two of the parent population from which the sampling is drawn, as well as the internal generalizability, whether the finding from a particular study can be generalized or inferred to the other setting or context beyond the sampled one. There is also the theoretical sampling method, which draws the theoretical propositions or principles statement from the finding of a study for more general application in future studies. We'll be taking a 15-minute break for Salat al-Asr and then continue our discussion. Assalamu alaikum wa rahmatullah. Welcome back, everyone. So after discussing the design of qualitative studies in specific, we will continue our discussion with the implementation of a research project. So some of the techniques or methods for data collection include observations, which consist of observing a phenomena in order to gain insight into how and why things happen. And meaning, for example, why a certain disease develops or why a certain complication happens. So this can include direct and indirect observation by a human observer of a certain phenomena in order to be able to describe it and to help to contribute to its understanding. So it is based on real events and its actual behavior that is observed is not verbal response as in questionnaires. So a major advantage of observation is its directness in terms of directly observing the intervention impact, for example. So in the observation, we don't ask people about their views, but rather watch them and what they do and listen to what they say in a certain condition. So it is rather a labor-intensive method, and the processing and interpretation of the acquired material is difficult and time-consuming in most cases. So observation is used, and especially in descriptive research, where there are many open questions that we need to observe for. So it can also be used in experimental research, where even when the researcher has a specific expectation about the result that is to be assessed. So observation is important to define precisely the things that need to be observed, such as using observations, schemes, which should be reliable and structures. For example, the system of categories where one can record the results. Also, it needs to establish that observation is the most suitable method of data collection in most cases, as well as establishing or working with several observers recording the same behavior by using audio-visual recording equipment so as to find the pattern of the behavior that we are investigating. So we need to ensure that the observed are unaware of being observed. This might affect their response or the way that they react to certain conditions. Also, we need to carry out a pilot study to check whether the method worked as expected or not. And observation methods could also include reviewing the record, observing signs, weighting, measuring height, and checking temperature. All of these are things that we can record or observe. So, questioning or interviewing facts are relatively easy to get at with these conditions. Also, there is some lapse in memories or response bias of various kinds, such as age may be related to certain conditions, as well as the disadvantage of that this method will have the possibility that the researcher will direct the subject toward a certain answer depending on how they react. In addition to this, the perception of the respondent in assessing the questionnaire, which is their framework of the aim of the research and of the answer that is desirable. For example, information given during a medical examination for a new job. So interviews are kind of a conversation, and that has a specific purpose. Also, it is initiated by the interviewer for the specific purpose of obtaining research relevant information and focusing on research objective in most cases. So interviews are considered to be flexible and adaptable ways of finding things out. It will have face-to-face interviews that offer the possibility of modifying one lines of answer of inquiry as well. It will also be time consuming, and anything that is less than 1 hour will be regarded as insufficient or invaluable. Also, anything that is over 1 hour may be more unreasonable or have demands on a busy interviewee, therefore can affect their response. So asking can also include conducting focus groups, discussions, administering written questionnaire, anything that can be included within these factors. So it is important to note that data could be collected using more than one technique that will depend on the participant's own views or perceptions. So data collection tools are the tools that are applied in data collection, and a particular technique can involve different tools to get at the desired answer that we are seeking to assess. So tools can include, for example, weighting scales, measuring tapes, microscope, observation checklist, some forms or questionnaires that we are assessing. So forms are used largely to record factual information such as the subject age, blood pressure, or treatment group. Also, forms are commonly used in clinical trials to follow a patient's progress and are often completed by the responsible investigator to obtain most accurate information. So the principles of questionnaire design include the content, in which the questionnaire should be designed to ask the minimum amount of questions, with each one justified in terms of the objective of the study and what can be gained from this information. So type of questionnaires include open-ended questionnaires. These allow the respondent to answer in their own terms and are used for non-categorical variables such as age, date of birth, and they can take more time and effort to complete. There is also the closed questions. These reduces the possibility of the interview's interpretation and coding bias and can facilitate processing. These can simply be answered by circling or picking a certain answer. There is also the closed-ended questions. These reduce the possibility of the interviewer's interpretations and coding bias that will facilitate the processing and can simply be answered by circling, as we have mentioned, and in closed questions where only one out of selection response is allowed, and the final option should always be if others please specify to include any missed information. There is also the Likert rating scales. These, for example, assessing or choosing the most appropriate answer, as well as wording of the questions, in which the questions must be written in a simple non-technical language, and the wording should be or should avoid any suggestion that a particular answer is preferred. There is also the questionnaire layout, which include an introduction and explanatory notes and instructions with examples of how to complete a particular question that should be brief in order to avoid including too much information. Also, the method of administration of the questions, which includes questionnaires that can either be self-administered, interviewer-administered, or computer-administered, and each one has its own advantages and disadvantages, with the computer one being the best and need less time to administer in most cases. There is also the method for administration that includes questionnaires that can either be self-administered or interview, with the question sequencing that should follow a logical sequence, following as far as possible, with the sequence that the respondent might expect to see with this type of questionnaires or answers. So recording of the response, the objective is to obtain a clear answer to each question, and it is important to ensure that when we are using closed questions, all the possible responses are included in a list of options that are provided. So failure to do so can result in biased response, thereby reducing care, incompletion, and incorporation. There is also the coding of the information, which is important from the beginning to decide on the coding scheme to be used for each variable. This will help to ensure that appropriate variables are being collected for analysis, and the coding guide should be constructed, which will provide the key to the meaning of the response. So some general considerations include that questionnaires should be subject to pre-testing and piloting in order to ensure that we have collected the most appropriate type of questions or included them in our questionnaire. Also, the assessment of the validity of the questionnaires in terms of whether all questions are relevant or some are extra questions. So it is desirable to use standard questionnaires, and they will usually have been used and proved satisfactory in use and may have been validated and will facilitate comparison of the results from your study with the data collected by other workers as well. So after the selection of the study variables, the study design and sampling method and data collection, a plan should be put for the data collection. This plan should include the following, including the type and number of data collection tools that is needed, such as questionnaires, as well as the categories and the number of data collection personnel, such as scientific technician, nutritionist, physician, whichever that we need to include in these type of information. Also, the duration and the content of training that is needed to be included, as well as scheduling or timetable for the data collection methods or tools needed for each phase, with logistical support such as vehicles, food and drink, and whatever that we need to include in order to have the best study outcome or data collection method. Also, we need to have an estimated budget with budget for tools and for training and whatever that we will be needing to conduct. Also, the plan for processing and analysis of the data must be prepared before the data is collected in the field so that it is still possible to make the necessary adjustment and anticipate for future events in general. So this is necessary so as to ensure that all the needed information is going to be collected in a systematic and standardized way. And this will also avoid the collection of unnecessary data uh, that will have too much data collection and will avoid any waste of the research. So sorting of the data with the data collation, making a careful comparison between the copies of the collecting sheets to learn the differences between them and to put likes with likes. For example, if there are more information with the questionnaire, putting each one separately, or cases with cases and controls with controls. In addition to this, performing quality control checks at every step, which is known as the data cleaning. This can be helpful in each step that could check for any missing information, as well as range check or even interviews that must be trained to follow a standard procedure, since the variation in the interview technique will lead to some sort of bias. Also, for the data processing, this could be either manually or computerized, and both can involve coding, which is the translation of the data into symbols that are appropriate for analysis. So this is true for the analysis of qualitative data in specific, and we should also aim to prepare for tables to be assessed. So the content of the research plan should include decision on the sequence in which tables should be analyzed, also estimate the total time for the analysis and decision on staff needed for the analysis, as well as estimate of the total cost of the analysis. So when we format the dissertation proposal, the title should be sufficiently detailed to inform the reader what the student or the researcher is proposing to do, with a maximum of 2 lines. Also, it should include a researchable problem in general, and these can be specific, population specific, place or date specific. After the title should come the introduction, which should include a selective review of the background to put the dissertation research into the context of appropriately recent literature. So this should include key research reports, a review of articles rather than textbook in about 2 to 3 pages, with outlining the problem to be assessed, as well as issues or topics for the dissertation and the need for undertaking research, this particular research project. Also, the research question should include a statement of the purpose of the proposed research in the form of a certain objective. So the hypothesis is not included in the descriptive study design, but rather should also be stated in this section, with the objective including the general ones and the specific type of objectives. And for the methodology, this should show how the researcher will address the research questions, and it has to include the design issue, such as the study type, which is analytical study, for example, a case control study. So all of these will be different depending on what we want to study or what we want to assess. Also, the time direction should also be included, whether it was a retrospective study or a prospective one, for example. It should also include the data gathering method or technique that was used. These can include identification of variables and how will they be measured, as well as providing a justification for the method that is selected, such as questionnaires or observation or controlled study. So all of these will be different depending on the type of study or collection method that we have used. After that should come the sampling technique and the nature of the population, the sample size, the selection of participants, all of which should be included in these steps. In addition to this, the techniques, the tools, and equipment that were needed should be included, followed by the resources and the references and the timetable for the study that is to be identified. Following that should be included the ethical issues that were faced in the study and the word count of the study that is to be proposed. And generally in the proposal, it should be between 1,000 and 1,500 words only. So after discussing the qualitative studies in more details, we will continue with the mixed method research and the study designs for them. These include the clinical practices, including diagnosing a disease or treating a disease, as well as preventing diseases from developing. So clinical research is a research that is conducted to improve any of the previous things, which is diagnosis of a disease, treating a disease, and preventing a disease. So there are main types of clinical research, which is the clinical trials, that is research to improve clinical practice, as well as experimental medicine, which is an experiment where a human is the subject, as well as the translational research, in which this is bench and bedside to back type of research to be conducted. So in vitro type of studies, these are cells in a dish and can be manipulated, but in vivo environment is lost in this type of studies. Therefore, treating a cell is a long way from treating a patient and cannot be regarded as the same or accounted to be as the same. Where in animal model studies, these can include or can sacrifice the animals and look at the tissue alone. Also, animals are not humans, therefore the result cannot be directly reapplied to humans, as well as animal models may partially resemble human disease, but the gene is not the same as in the actual condition. There is also the dead bites of animals. These can include tissue banks and biopsies. It is the correct subject, but it is, and also the correct disease. So only one time point to be examined in this case, which is normally the end, and they have died and no intervention is possible to be applied within this condition. Where in living people, these are experimental medical studies and clinical trials. We are studying the true disease using the correct subject, and intervention is possible within these cases. But these are very limited in what we can measure and also very expensive in terms of what we can assess, therefore can be conducted only with minor uh, possibilities or alterations. So in epidemiology studies, these can include, for example, identification of certain genes or even drugs that will only have clues of what we want to assess rather on the exact assessment point that we can consider. So the drug development stages can include at least 10 to 12 years for a drug to be approved or for a medicine to be approved for human use. These include the drug discovery period, the preclinical trial period, the clinical trial phase, which is divided into 3 or even 4 phases. There is the regulatory review and the scale up to manufacture and the post-marketing assessment. So during this phase, particularly the clinical trials, not much of the study can proceed or go on to the next stage, but rather are very difficult to obtain. So these include high iteration rates, and most studies fail for most reasons include commercial strategies, their efficacy, and their safety procedures. So how do we improve the decision making about which medication to develop? This is a complex question with a complex answer as well. But we all agree that we need better translational medicine as well as clinical research, experimental medicine, or whatever we need to call, or not relying mainly on mice for studies or for assessment. So some of the tools that are used in experimental medicine include biobanks, which are human tissues repositories, as well as imaging facilities, genetics, and clinical research facilities. So biobank, we study whether the disease express the target of the drug, which is the biomarker or target validation. Also, it may include tissues, not just blood, but rather need rigorous annotation, linkage to clinical trials and data with appropriate governance procedure. This is an example of a postmortem brain from a patient with multiple sclerosis, as we can see in the auto diagram with radioligand that finds the macrophage protein. So in imaging, we can use the pharmacokinetic, which will detect and quantify the drug itself or the drug target. This will help us in assessing whether the drug gets into the brain and what dose do we need and how long does it last in there. In the pharmacodynamic, this will help in detecting and quantifying the markers of the drug effect. We can assess whether the drug do anything, and trials for chronic disease are long, large, and therefore expensive. So it would be nice to have an indication that the medication might actually work and the molecular changes that precede the structural ones, which precede the clinical trial development. So using PET scans in the receptor occupancy studies can show the expression of certain receptors and can be helpful for studying the dose of the medication that is needed. Also, MRI assessment of RV structure and function can be helpful. And this is an example of facilitating research that is theoretical. It doesn't have to be high-tech, but rather exploratory type of studies that we can assess. So the study design, for example, can include 6 men and 6 females with age between 26. The normal weight or BMI should be 24, with assessing the markers of the BYY versus the saline, with a crossover study and a double-blinded one with randomization. So in this method, we can assess, for example, their feeling of hungry and how this would relate to their medication being intake. So when we want to initiate a clinical trial, we need to define the questions and design the study by obtaining scientific peer review, secure funding, and prepare for the study document when it is needed. This is an example of a toolkit for the data and tissues to be included. And there are differences between absorption and the distribution of the medication. These will depend upon the stream that is to be observed, including, for example, the oral drug will be absorbed in the bloodstream, and we can measure the connection in the blood or concentration with the blood that is delivered to the certain route. Also, this will be followed by the distribution in the site of action, or even lost for other places as well, in which this can include the elimination of the drug. So membrane, the plasma membrane and the lipid bilayer can encapsulate the cell, and the basement member will include the base layer of the cells. So passive movement of the drug across the membrane can be different. For example, for propranolol, these can cause nightmare, but atenolol doesn't. This will include to the drug absorbability in the water or lipid solubility. So this is due to the effects of how they move across the cell layers, as the lipid-soluble lipophilic often cross the plasma membrane, while the water-soluble, which is known as the hydrophilic, often doesn't. Therefore, this will be related to their absorption effect. So the organs that have the tightest barrier is the brain cell membrane layer, while the weakest one is the renal layer. So this graph can show the permeability increasing as the size decreases, while the permeability also increases as the lipophilicity increases. So size is more important, and we can compare the medication absorption depending upon the molecule size. And for the active transport, these can include the beta-glycoprotein, mainly in the gut wall and the brain as well. There is also different drug responses in patients with B-glycoprotein mutation, for example, within these cases. So the bioavailability is defined as the percentage of the drug that reaches the systematic circulation. So all medications have about 100% bioavailability when they are given as an IV injection. But rarely there is 100% when the medication is given via an oral route, therefore its observability is reduced. So this can be due to the destruction of the medication by the stomach acid or bacteria, as well as if the medication was too big or hydrophilic to easily cross the gut wall. So the bioavailability is the percentage of the drug that reaches the systematic circulation. An example of this is if we take 1 gram of paracetamol and only 800 milligram will reach the systematic circulation. Therefore, we can say that the bioavailability of this medication is 80%. So this is an illustration of how the bioavailability of medication is different when it is given via an IV route where it was given by an oral route. So the variability in the bioavailability will depend upon the stomach pH values, and it is highly variable throughout the day and can increase by food intake. Therefore, when we are given some medication, we can be told to consume it with food or after a meal. Also, the exposure to the stomach pH, which can include reduction in the gastric emptying in general, as well as relation to food or the drug mechanism and the certain disease conditions, such as gastroparesis. Also, the CYP enzyme in the gut wall and the liver can affect the drug absorption and the transporters such as the B-glycoproteins. So as we have mentioned, absorption is highly variable and it matters depending on the drug percentage that we aim to absorb. So absorption and distribution is mainly a passive process, and the membrane factors can include gaps between the cells, and the drug factors can include the size and the lipophilicity or the charges of the medication. So bioavailability as well can be highly variable, but it doesn't always matter depending on the condition of the patient. There is also the volume of the distribution that help in providing clues to where the drug has gone and determine the half-life of the medication. So an important consideration in medication intake is the renal elimination. So the glomerular filtration rate plus the secretion minus the reabsorption will give us the urine output, which can determine the medication general absorption in the circulation. So glomerulus is our leakiest membrane, and all drugs will have that is less than 5 kDa in size will be filtered regardless of their charge or their lipophilicity, except for the ones that are protein bound. So for 125 millibare a minute, the entire plasma is circulated or filtered in 25 minutes, and a full bladder is developed within 3 minutes. While in the tubular reabsorption, this occurs in the loop of Henle and the distal tubules, which is the tightest cell layer of the renal parts. These will be sodium actively pumped from the filter to the plasma, and the water will follow by a process of osmosis. So the loop of Henle and the distal tubules is the very tight cell layer of the paracellular that is not available. Also, it will have ions pumped back, which is transcellular, and almost all of the water reabsorbed in general. So the drugs are highly concentrated infiltrate at this phase, while the hydrophilic drugs are trapped and eliminated in urine, and the lipophilic drugs are escape or return to the blood to be absorbed by the circulation. So drugs are eliminated either by the liver or by the kidney. If they are lipophilic, they will be eliminated by the liver, and if they are hydrophilic, they will be eliminated by the kidneys. So renal impairment can affect the drugs in three ways. These include reducing the renal clearance of the drug, also reducing the renal clearance of the drug metabolite, and extrarenal effects of the renal impairment that will lead to a major impact on the cells being reabsorbed. So uremia will reduce the expression level and impair the function of the metabolic enzyme, such as the CYP enzyme, and the transporter molecules, which will result in more drug being absorbed from the gut and less metabolite uh, being absorbed from the gut and the liver everywhere. So the glomerular filtration rate is only part of the renal elimination, but it does predict the renal drug handling in general, and it is quantified by measuring creatinine as a natural drug. And the options are to reduce the dose in general for this type of medication or increase the dose interval to ensure for a better absorption in general. So the kidneys, in summary, will eliminate the hydrophilic drugs, but cannot eliminate the lipophilic drugs. Also, impairment will reduce the elimination, but also reduces the metabolism of the lipophilic drugs via indirect effects. So another important consideration when we are conducting clinical trials for investigation and medical products include the half-life and the clearance of the drug. So the plasma half-life is the amount of time that is required for the plasma drug concentration to half into half. So this will be their unit of time with the same percentage that is removed, but not the same amount. So some drugs will have less than 5% worth of differentiality, and they don't have a half-life to be filtered. So the plug hole, which is the first order, versus the bucket, which is the zero order, is a method that we can use to assess the drug absorption or the drug half-life. So the plug hole kinetic is the amount that is eliminated proportionate to the drug concentration, for example, most of which can be eliminated. Where the bucket kinetics is the amount eliminated in constant concentration that is irrelevant to the drug amount. So the amount of time for the biological effect of the drug to half into half is that we don't actually formally measure this, but we need to consider it because it will affect the clinical trial design, such as the crossover trials and when to expect the outcome. So the clinical question, for example, to assess a man who is accidentally taking 4-time dose for their normal atenolol dose, they would feel dizzy and their pulse would raise or decline, which is he's admitted to the hospital. And the question is whether or not he can be discharged the next day. So what we know about atenolol is that the half-life is 6 hours, and therefore this man can be discharged. So we need to understand the three most conditions that are met, which is the concentration in the plasma related to the site of action, also the drug binding the target in a reversible manner, and the effect that relies on the drug that is being there. So the plasma half-life is 15 minutes, while the biological half-life for aspirin is a few days. So why does the plasma half-life is different from the biological one? This is because the fact that it binds in a reversible manner, and the biological half-life is often longer because of the platelet factors that have no nuclei and can live for about 8 days. So the plasma half-life doesn't change with the indication, but the biological half-life might. This is due to the platelet inhibition and the side effects of the medication, as the immune cells, for example, can synthesize certain replacement in some cases. So atenolol half-life, for example, is 6 hours in the previous example, and the half-life, as we said, is 6 hours, then therefore this man can be discharged the next day because they have cleared their intake. So the clearance will depend upon the volume of the plasma that is cleaned of the drug per minute and is expressed as millis per minute, and most medication are cleared by the liver and then eliminated by the kidney from the body. So in summary, the plasma and the biological half-life are not always the same, as the biological half-life can depend more on the system than the drug, and the same drug may have different biological half-lives depending for each effect. Also, half-life is only relevant concept for the first order kinetic, and the determinants of the half-life are the volume and the clearance rate. So the adverse drug reaction is that all medication, even if they are used correctly, can have some potential adverse effects, as every prescription requires the risk-benefit analysis, and some tactics are very rare adverse effects, with some are explicit, such as if you have peripheral neuropathy, then some medication can have an adverse effect on your health. So this is defined as effect of a drug that is undesirable and uncomfortable or even dangerous, and can occur with a normal use of the medication, and is considered distinct from toxicology, which is the overdose that is increased of the plasma level. But this can blur for the drug with a therapeutic index as leading from warfarin intake. So about 5% of hospital admissions are due to adverse drug reactions, and 50% of the hospital admission involve the development of adverse drug reaction, in which 15% of them can be life-threatening. So who is at risk of developing adverse drug reaction? These include elderly in specific because they have polypharmacy and pharmacokinetic impacts, as well that most trial only involve young patients on a fewer drug that is being taken. So the top three classes of medication that cause adverse drug reactions are warfarin, aspirin, and non-steroidal anti-inflammatory drugs, as well as diuretics. So all of these have adverse drug reaction in most cases. So the classification of medication is related to the dose adverse reaction, whether it was considered as an allergic reaction or as idiosyncratic due to no obvious cause, but rather as the patient's own response or reaction to the medication. So there is a clinical scale that is used to assess the adverse drug reaction, and the score can range from 1 to 4 depending on the severity of the drug reaction that is to be assessed. So the severity can be regarded either as mild, moderated, severe, or in some cases as lethal cases for the medication. So, for example, constipation, UTI, sepsis, and death, these will be regarded or ranging from mild to lethal cases. So, the allergic hypersensitivity reaction will be drug binding or relevant immune cell proliferation, depending upon the factor that is to be assessed. So some of the common adverse drug reaction will be detected in clinical trial, which is the ideal timing, but about 10% will be only detected after the medication has been approved and distributed. So identifying the adverse drug reaction in specific patients will depend upon the chronicity of the medication. This will be immediate, relatively easy, or delayed, much harder impact. So we can stop and re-challenge in these cases, and the drug can be stopped, for example, or can the reaction be allowed to happen again, depending upon the severity of the condition that was developed. So when these cases develop, we need to either stop the medication, reduce the dose, or even switch the class of the medication to one that is more easily absorbed by the patient. So in the first administration to humans studies, we need to understand why do we need before the study is conducted, also the objective, the responsibility, as well as the risk assessment and minimizing these risks, and the study population, whether they are healthy or patient volunteers, as well as selecting the appropriate dose and study design in most cases. So the objective for first in human study is to assess the tolerability and the safety, as well as the pharmacokinetic in the relevant dose range, and the pharmacodynamic effects, as well as the dose concentration response for the desired and undesired side effects. So what do we need before the first in human trials is to have satisfactory evidence of the desired activity, also the toxicology to be assessed, the satisfactory safety of the pharmacology, as well as the pharmacokinetic effects and the metabolism, including studies of metabolic enzyme inhibition and induction. Also, the sensitivity and specific validated assays in human and the pharmacoceutical formulation from a batch of a known purity and stability manufactured of GBA. So who is responsible for conducting all of these steps? Is that many people involved in the first in human trials, but the one who's responsible for the safety could be the sponsor, as well as the organization conducting the study, the principal investigator, and the nursing or the researchers with the ethical committee or the regulatory authorities. So to minimize these risks, we need to have the investigator brochure, which is in the preclinical data that suggests that there will be a useful therapeutic window, also to enable the data to calculate the starting dose with reasonable certainty and what are the likely first signs of toxicity and whether they are difficult or easy to assess. So the high risk of investigational medical products can be with the biological or gene therapies in specific. These can contribute to the risk by assessing the quality of the material that is used, as well as the non-clinical test for the safety and the pharmacokinetic of the questionable relevance, as well as the expression of the targets on other off-target cell types or tissue that may result in an unexpected biological activity in humans, in addition to immunogenicity that can result in hypersensitivity reaction due to activation of certain pathways. So to minimize the risk, we need to assess the study design by using the preclinical information to be selected, also the study population to be assessed, the number of subjects that are recruited, and the starting dose with the dose increment and the maximum dose to be included. Also the safety parameters to be measured, the duration of infusion, the parallel versus crossover design, the intervals between the dosing of the subject, as well as the duration of observation and the washout interval before the dosing, or the same or the different impact. So when we are choosing the principal investigator and the staff appropriately who are qualified, experienced, and equipped to assess for adverse drug reaction whenever they are present. We should also use a lead sensitive volunteer and set a clear stopping rule in order to be assessed on a regular basis, as well as recommending an antidote and trained staff in its use. Also reviewing all the data before escalating the dose and be prepared to modify the design in light of the data in general. So the study population will have advantages of using healthy volunteers. These will be readily available and providing rapid recruitment rather than specific impacts. Also, there will be no confounding factors such as disease or other medication intake. Also, it can usually tolerate pharmacological insult and can accept intensive scheduled procedures and can freely give informed consent in most cases. So, what is a healthy volunteer? They are defined as an individual who is in good general health and not having any mental or physical disorder that require regular or frequent medication, and who is also able to give a valid informed consent to participate in the study. So we could learn more by obtaining information in patient if it is practical or even safe to be included. So in patient expressing the target medication, the biological may show the target-mediated drug of action or disposition. Also, the tolerability that may be quite different from that in subjects who don't express the target, and some drugs are too toxic to be included in certain studies, therefore are mostly avoided. So the selection of the study population is first we should aim to do no harm, minimize the risk, and have minimal risk. We can include healthy volunteers that shouldn't be exposed to anything more than the minimum risk. And in phase 1 study, this shouldn't be toxicity study, but rather to assess for the general. So for any medication, we need to choose the route of administration. For example, in small molecules, we only use oral intake because we should consider the variability in the bioavailability, the rate of absorption, and therefore the Cmax and the Tmax. In addition, we should assess for the taste and the blinding, and there is no going back once the medication is administered via an oral route. Also in the intravenous, these can also be small molecules and biologics. These can be administered slowly, and we can slow down or stop the infusion if there are any adverse effects that have developed. So choosing the dose will depend upon the reasonable starting dose that is to be administered. Also, what is the maximum dose that could be tested? And what are the reasonable dose increments until we reach our desired dose or interventional dose? In addition, how many doses can reasonably be tested as to assess for the maximum absorbed dose, as well as what are the stopping endpoints in terms of assessing adverse effects, that should we dose to toxicity and the plasma concentration of the medication, as well as the pharmacodynamic endpoint, which can include the maximum enzyme inhibition and the receptor blockage or the receptor occupancy. So according to the FDA, the maximum recommending starting dose should be first determined with no observable adverse effect level, which is known as the NOEL. Also for step 2, we should convert the NOEL from the most appropriate species to the human equivalent dose in most cases, as well as selecting the human equivalent dose and taking into account any additional factors, including the metabolism or the receptors, and applying a safety factor to give the maximum recommended dose to be tested or assessed in these cases, as well as giving these maximum recommended starting dose based on organs assessment. So the basis for dose selection include the pharmacology of the medication, in which the range of the dose at which the desired effect is seen in vitro or in vivo studies, as well as the animal models and the relevance of those models. Also, the range of the concentration at which the desired effects are seen in vivo in animals models and the relevance of those models. So this is the dose response curve in which we can assess the maximum absorbable dose, the ED and EV factors, and the potency of the medication. So for pharmacology, the range of the doses at which the desired effect is seen in vitro or in vivo in the animal models and the relevance doses to those models can be assessed, as well as the range of concentration at which the desired effect is assessed, and the initial target to be assessed in most cases. So the nature of the pharmacodynamic activity in animals should also be assessed, with the slope of the dose response curve to be also included or assessed in these studies. And the secondary pharmacology should be assessed with regard to response on the CNS, the CVS, and the respiratory system as well in most of these cases. So all of these should be assessed to determine the most appropriate impact on the patients as well. So it is to be expected that the dose reducing the undesirable effect in the animals will be lower than those reducing the target effect on the organ toxicity. So for the toxicology, these can include the plasma concentration of the medication and the exposure with the maximum no adverse effect level done well. That is most sensitive toxicology species can assess in most cases. So the nature and the severity of the toxicity that is seen will also assess the predictability of the pharmacokinetic of these medication in, from the animal models. So the necessity or the consequence of demonstrating toxicity can be assessed in these phases of the medication intake. So in a single ascending dose study design, these should be randomized in order to ensure for the best outcome, as well as should have a placebo-controlled double-blinded type of study. So this can be helpful in allowing for the intra-crossover and inter-individual assessment of the studies, as well as the comparison of the adverse drug reaction and the pharmacodynamic measures. Also, this will be helpful in avoiding the development or the bias in conducting the study and the attribution, in addition to the handling and interpretation of adverse drug reaction or effects, including the lab data. So the statisticians can generate codes at this phase in order to ensure for the best outcome, as well as the coding that can be made available in individuals who require it during the study, such as the bioanalyst in general. Also, the code can be broken for any individual doses if it was assessed for the safety or for ethical responses to ensure for the best outcome. So each subject in this case participates on one occasion and only on a new cohort that is treated at each level, as well as each subject can participate on several occasions with the same cohort treated at various doses level using a crossover randomization method. In addition to this, each subject can participate on several occasions with the same cohort that is treated at various dose level using a parallel group administration method, depending upon the method that we aim to assess or we aim to achieve. So this is an example of how to develop a cohort of medication intake that will have advantages of almost no problem with the dropout rate, the blinding, the carryover that is due to long T-life or half-life of the medication, and limiting the blood volume sample with disadvantages that it can be uneconomic with many volunteers that are screened, and also no possibility for intra-subject comparison of the pharmacodynamic and dynamic as well. So in these cases, randomization is restricted to prevent any subject from receiving the placebo on successive occasions more than one time. So in the crossover design, this can have advantages such as economic advantages with maximum information from small number of subjects, as well as that randomization at each dose level provide within the subject conversion of the adverse drug reactions, as well as the within the subject conversion of these factors. So a sufficient doses for intra-subject assessment of the linearity and the variability of the pharmacokinetic or parameters can be assessed in these cases. Also, it can add a feature, a counter period of crossover at a selected dose, therefore providing within subject conversion of the adverse effect of the medication and pharmacodynamic measures at any dose, and can increase the number of exposed to that dose level. While the disadvantages of this method include that if the study stopped early, then some subjects will not have received the placebo, and the amount of the blood taken from each subject may limit the number of exposure. Also, the washout period may need to be extended if the half-life is longer than expected, therefore making the study protracted with a possibility of a high dropout rate. There is also the parallel single study dose design, in which this will have parallel group and each subject can participate on 4 different occasions. The advantages of this method include no omission of the dose or complication from stopping early on, and the linearity of the pharmacokinetic that can be established within the individuals, while the disadvantages can include much more difficult to maintain the blindness and no placebo or controlled intra-subject conversion of the pharmacodynamic to be assessed. So how many subjects or how many doses can be included in this design? This will be based on the decision for the dose escalation or the selection of the dose for the next study. Also, to be sure about the linearity or of the pharmacokinetic and to give a reliable estimation of these pharmacokinetic parameters and to give a reasonable estimation of the pharmacodynamic measures with meeting the study objective without putting unnecessary number of subjects at risk. So the dose intervals for this type of study, we need to ask the questions of how many subjects should we dose on the first day at a new dosing level to reduce the risk by staging the exposure. Also, we need to ask what intervals should we leave between the dosing different subjects at a particular dose level to ensure for the best outcome, as well as to ask what intervals should we leave before administering the next dose level with the washout period in general. So this is an example of a biological study, first in human study, with combining single and multiple doses during a duration of 1 year divided upon the total number of participants. So what else might we include in a first in human trial? These can include the fed versus the fasting status, as well as the elderly or the young conversion of the pharmacokinetic and dynamic, also the solid dosage, the tolerability, the safety, and usually this can be conducted within a separate study design. So the data from an exploratory studies can include the data on tolerability and safety in general, as well as the adverse events and adverse reaction in details. Also the pharmacokinetics and in the relevant doses range, as well as the parameters, the dose proportions, and the concentration on all of these cases with the pharmacodynamic activity, including the dose concentration response and the relationship between the pharmacokinetic and dynamic. So in conclusion, these include the safety of the volunteers, that is paramount, and we must always minimize the risk, but anything more than the minimum risk is unacceptable for healthy volunteers. And there are a variety of study design that are suitable for first in human study with the risk that can be minimized by taking into account the nature of the molecules and what we understand about its mechanism of action, as well as using the most appropriate study design and the rationale for selecting these designs. Also, the pharmacology with ensuring those conducting the study are trustworthy and qualified, as well as for monitoring all the safety pharmacokinetic data during the study and modifying the procedure if necessary. Also, to stopping when it is sensible to do so, because we need to ensure the safety of the participants at all stages. So an important consideration when we are conducting research or health research is the quality assurance and the quality control in clinical research. So quality in clinical research, we will focus upon defining quality, the systems and the control of the quality, as well as the assurance and the responsibility with the TMFs and site files or inspections in general. So quality is defined as the scientific integrity. These can include robust protocols that are accurate and reliable data. Also, it should have clear specific aims with scientific questions that are being modified, and our written questionnaires or research question with no ambiguity. That should also be comprehensive, meet the legal guidelines and requirements, with funded and resources that are ensured, and should have sensible recruitment. So quality should also include having trustworthy procedures with standard operation procedures and reliable, well-maintained equipment with no unnecessary risk to the participant, as well as safety procedures, medicine safety, and reasonable burden on the participant, as well as having trained staff and informed participants in all cases. So quality control is defined as the operational techniques and activities that are undertaken to verify that the requirements for the quality of the trial-related activities has been fully fulfilled. So in practice, this includes monitoring, management oversight, steering the committee, the data safety monitoring committee as well. While quality assurance is defined as all of these planned and systematic actions that are established to ensure that the trial is performed and the data are generated, documented, and reported in compliance with the good clinical practice. So quality assurance can include in practice audits, SOPs, policies, templates, and inspections. So quality assurance can make sure that quality control is effective. And according to the GCP requirements, the systems with procedures that assure the quality of every aspect of the trial should be implemented. And legal requirements for investigational medical product trials and guidance for all other clinical research should be included to minimize errors and detect for any mistakes. So why do we need quality assurance and control? This will help to have or reduce a poor quality trial that is a waste of time, as well as money and resources, and can include unnecessary burden to the participants as well. Therefore, it is unethical. So the sponsor is responsible for implementing and maintaining quality assurance and quality control system with written SOPs, and the sponsor should review systematically with well appointed monitors and auditors, as well as they may appoint quality assurance manager and governance lead, and will delegate, therefore, everyone involved in conducting the trial that must take responsibility for their own work. Also, quality in the staff can include recruitment of qualified, experienced staff, also training them on appropriate procedures and protocols. So the protocols can include having peer review, expert input, and science report, as well as statisticians that are to be implemented procedures. So these procedures include SOPs, manuals, and protocol checklists Also, it can include equipment calibration and maintenance with appointing appropriate suppliers. And we should have a high quality investigational medical product to ensure for good medical practice and quality product release. So having a standard operation procedure will include a detailed written instructions to achieve a uniformity of the performance on a specific function. So a clearly written description of how a particular task is to be performed by who and who's responsible and when and where is needed at all steps. So this can be used to train staff and to help to ensure the consistency, the compliance, accountability, and efficiency of these medications to be integrated. So a standard operation procedure includes typical section of cover page with authorizations, also introduction with background, as well as the purpose with the scope and the responsibility and the specific procedures to be followed with the associate documents or form, as well as reference and change in history, whether it has been conducted or not. So for the standard operation procedures, there are numerous examples available online that we can assess and study in order to have the best outcome. So audit, which is about quality assurance or control, include a systematic as well as independent examination of the trial to evaluate that activities were conducted, the data recorded, analyzed, and accurately reported according to the protocol, sponsors, and the GCP, which are applicable regulatory requirements for all cases. So for quality assurance, this is a part of audit that is independent snapshot examination by a trained auditor, not by the monitorer. And the processes include documentation of the data and the investigator sites with vendors and laboratory or key documents being assessed. So audit is considered to be systematic and independent review, as well as audit that is against the standard GCB, the protocol, and SOB to be assessed at all cases. So monitoring is considered to be a part of quality control. These include the act of overseeing the progress of a clinical trial and ensuring that it is conducted with the progress and overseeing or ensuring the conduction, the recording, and reporting in accordance with the protocol. So the purpose of monitoring is to ensure that the clinical trial results are reliable and the treatment effects are real and unbiased, but further information in detail will be covered on later. So the GCP inspection is conducted thorough review of the document, the facilities, as well as the resources, and can inspect the sponsor or the host organization, as well as a service that is being provided, such as laboratory setting. And inspection may also be routine or triggered or even MA driven. So some of the common findings in non-commercial studies include critical and major inspections, including the trial management, the quality system, the monitoring, or a serious breach in general. So finding an inspection can impact the organization and its reputation. It will also be regarded as a criminal offense that start or conduct trial without an investigation or recommendation product and fail to notify the end of the trial or having any serious breach. So some of the frequent inspection findings include that IT systems wasn't sufficiently validated or insecure, also poor medical oversight, insufficient SOPs, or transcription errors. So what is GCP? This includes the good clinical practice that is defined as an international ethical and scientific quality standard for designing, conducting, recording, and reporting the trials that involve the participation of human subjects. So this will provide assurance that the data and the reported results are credible and accurate. Also, assurance that the rights and the confidentiality of the trial subjects are protected at all costs. So the GCP is consistent with the principles of the Declaration of Helsinki. Also, the unified standard that provides mutual acceptance of clinical trial data by the regulatory authorities and can provide harmonization of the regulatory authorities and area of the safety, quality, efficacy, and multidisciplinary impact. So the International Conference on Harmonization is an organization of regulators and pharma industry to discuss any scientific and technical aspect of product registration in general. So a clinical research is any research that involves human subjects and can include clinical trial using medical product, questionnaire or interview studies, as well as research on biological sample or tissues and healthy volunteers or health system participants. So the GCB has developed based on tragic accidents in the past that were conducted in clinical trials, such as the Takuji syphilis studies, in which 40-year study has progressed a cohort study of 400 African American men with syphilis. They were denied from taking the medication even though if it was available. Also, this was followed by the incident in the number code in which a voluntary consent of the subject has been made mandatory, and that result should be fruitful and not procurable by any other means. Also, the trial design should be based on the result of an animal experimentation before it is conducted on a human subject. And every possible action must be taken to protect the subject from injury, disability, or even death. as well as that experiments should be conducted by scientifically qualified personnel and subjects may be able to terminate the trial at any stage. Also, in the thyroid tragedy that was in the 1950s and continued until the 60s, originally they were prescribed as painkillers and sedatives and widely prescribed as antiemetic for morning sickness and sedative. This has resulted in about 12,000 birth defects in 46 different countries. So, animal testing for birth defects subsequently was introduced. Also, the Declaration of Helsinki has been changed over the years. In 2008, the key changes including the underrepresented population that should be provided with access to participate, and inclusion of the standard information in the protocol with appropriately qualified individuals who are able to take consent. And in 2013, the key changes included increased protection for vulnerable participants and compensation for harm requirement, as well access to results and benefits post the study. So these are the historical events that has shaped the GCB as we know it today, and to ensure that the best outcome developed for the participant safety. So the principles of GCB include that clinical trials should be conducted in accordance with the principles of the Declaration of Helsinki, GCB, and its regulation. Also, the anticipated benefit for the individual and the society should always outweigh the foreseeable risk and inconvenience, and the right, the safety, and well-being of the subject should prevail over the interest of science and society, where the available non-clinical and clinical information on investigational medical products should be adequate to support the proposed trial, and the trial should be scientifically sound and described in a clear, detailed protocol. And the trial should also be conducted in compliance with the protocol, which has ethics committee approval and favorable opinion. So the medical care and the decision as well should always be the responsibility of the qualified physician. Another important aspect of the GCB include that each individual involved in conducting the trial should be qualified by education, training, and experience to perform their own responsibility or task. Also, they should be freely given informed consent that should be obtained from every subject before their clinical trial participation, and all clinical trial information should be recorded, handled, and stored so as to allow its accurate reporting, interpretation, and verification, irrespective of the type of the media that was used. So the confidentiality of the records must be protected in accordance with the regulations, and any investigational medical product should be manufactured, handled, and stored in accordance with the GMP and used in accordance with the approved protocol. So the systems that assures the quality of the every aspect of the trial should be implemented in aspects of the trial that are essential to ensure human subjects are protected and the reliability of the factors that are to be focused upon. So additional principles from the UK clinical trial regulation in 2006 amendment include that the rights of each subject to physical and mental integrity should be protected, and they must be safeguarded, and licensing authority approval is required with the protocol that must define the inclusion and exclusion criteria and monitoring and publication policy with the provision that must be made for insurance indemnity to cover the investigator or sponsor liability. So, what is a clinical trial of an investigational medical product? This is any investigation in human subjects other than the non-interventional trial that is intended to discover or verify the clinical or pharmacological or other pharmacodynamic effect of one or more medicinal products. Also, to identify any adverse reaction to one or more of such products, or to study the absorption, distribution, metabolism, and excretion of one or more of these products. This will be with the objective of ascertaining the safety or the efficacy of those products. So this is a list of assessing the investigational medical product in order to implement a clinical trial. So research governance is defined as systems or regulation that ensure the good practice and the high quality at all stages of the research process. So these will be helpful in ensuring that researchers do the right thing in the right way at the right time. Also, there is no new legalization to bring, but the existing regulation that is to be altered or to add guidance of clinical research into one document. So in the NHS research governance, the purpose of these are to ensure the dignity, the rights, and well-being of the public. Also, to improve the research quality, as well as to prevent the poor performance, and to help prevent the misconduct and the profound uh, fraud in these research projects. So the policy framework for health and social care that was developed in 2017 include focusing on patients, the service users, and the public, as well as having safer, more efficient, or more effective treatment, as well as care and other services to be developed and tested. Also, we should be applying to do research to be less barbaric, as well as the industry sees that it's a great location to be assessed for research. So the policy framework for health and social care include 19 principles, focusing on details for the chief investigators, the research terms, as well as the students and supervisors, as well as the sponsor or the research site and regulations. So the GCP cover as well the documentation, including the protocols, the trial records, the approvals, any amendments, and recruitment procedures, as well as the consent and safety regulation to be included in the study. So safety reporting is considered to be legal requirement as in the medicine for human use clinical trial regulations, in which an investigation or shall report any serious adverse event immediately to the sponsor, as well as the sponsor shall keep detailed record of all adverse events that develop into a clinical trial, which are reported to them by the investigator for that trial. So these can include the annual reports, and there are two types of annual reports that we need to include, including the annual progress report and the developmental safety report. So at the end of the trial, we should also have an end of study notification and the final report within 12 months of administration. So the annual progress report includes or should be submitted on the anniversary of the original ethical approval that was obtained, and should update the required ethical committee on the site information, the recruitment and amendment, as well as the safety procedures at all times. So the developmental safety update report has replaced the annual safety report to the MHR in September 2011, and it was introduced to improve the harmonization across Europe and the USA in specific. Also, it includes comprehensive annual safety review of an investigational drug or a multi-drug therapy, as well as this should be the sponsor responsibility that is delegated to the chief investigator in any academic study. So after the trial completion, we should have an end of study notification that is sent to the required ethical committees within 90 days, unless early termination, then it should be sent out within 15 days. Also, the final study report should be sent to the MHRA within 1 year of the end of study notification. After that should be archiving, in which the SOB on archives, as well as keeping a log of all documents and where they will be located, as well as ensuring that the data and any software is retrievable. Also, the label of the medical notes that to be retained, as well as keeping electronic records, and the need for careful archiving for these impacts. So how long should we archive these data? These will depend on the local requirement. For example, 10 years after the trial being ended. Also, at least 3 years after the last marketing authorization of the trial, if they contribute to that authorization. And if the trial involves children until the youngest participant reaches the age of 21, and at least 30 years for advanced therapy trials such as gene therapy or cell therapy. And it will be 25 years when the new regulations are enforced, and sometimes indefinitely depending on the method to be assessed. So the safety reporting is a legal requirement as the Medicine for Human Use clinical trial regulation state that an investigator shall report any serious adverse event immediately to the sponsor, and the sponsor shall keep detailed record of all adverse events relating to the clinical trial, which are reported to them by the investigator of that clinical trial. So the safety reporting is conducted to ensure the benefits of a new drug outweigh the risk and the side effects as well. Also, it will be the sponsor responsibility. And for the non-commercial study, the chief investigator is usually take on this responsibility. Also, delegation must be documented once it is happening, and the clinical trial regulation could include defining the type of adverse event and any timelines for reporting. So for adverse events, these are any unwanted medical occurrences in patients who are subject to administered a medical product, which doesn't necessarily have to have a casual relationship with this treatment. So this can be unfavorable sign, symptom, or even disease status, and should be recorded on adverse event form or log and the medical notes as well. Also, for the adverse event collection, these should be questioning the participant at each visit if they have experienced any side effects, as well as assessing for any abnormal lab results such as ECGs and medical notes and any admission or visit to other clinic or anything that was unexpected. So they should keep diary entries in order to assure that any adverse event is being recorded or reported. And for the adverse reaction, these are defined as all the unwanted and unintended responses to a medicinal product that is related to any dose was administered. So there is a reasonable possibility that the event is related, as it cannot be ruled out at any base, but therefore should be assessed. Also, the occurrence of any serious adverse event is defined as any adverse event that results in death, also is life-threatening, as well as requiring inpatient hospitalization or prolongation of existing hospitalization, as well as resulting in persistent or significant disability or incapacity, and is regarded to be a congenital anomaly or birth defect, as well as medically important event. So the severity of any adverse reaction will describe the intensity of that reaction. For example, mild, moderate, or severe, such as severe headaches. Also, the seriousness of these reactions, including severe serving as a guide of reporting, such as 6 scenarios that we have mentioned earlier. So, a serious adverse reaction is where a causal relationship between the medicinal product and the serious adverse reaction is at least a reasonable possibility. For example, their relationship cannot be ruled out, while the causality must be assessed by a medically qualified investigator at these steps. So the foreseeable serious adverse reactions and events often have certain events that can be predicted and can be defined in protocols as not required any immediate reporting or intervention, while the disease-related or pre-existing condition and population-related, such as an elderly or serious medical condition, the drug-related events, which are considered to be predictable. So the expectedness is if a serious adverse reaction is suspected or confirmed, then a nominated individual needs to determine whether it was expected or need to be appropriately trained, but not necessarily a medic. They should refer to the protocol and the reference safety information. If it wasn't expected, then this will be considered as a serious, unexpected, serious adverse reaction. So a serious expected reaction is the nature of the severity of which it is not consistent with the applicable product information, but rather unpredictable. So this must be reported immediately to the sponsor and to the required ethical committee within 7 calendar days if it was life-threatening, and within 14 calendar days for other serious unexpected reactions, and reported electronically for some authorities. So a serious unexpected reaction is an event which is not listed in the RSA or is listed but has occurred at an increased frequency or severity or in a different specificity. So, for example, if something is listed in RSA, such as hypertension, this will be suspected for a participant with hypertensive crisis, but unexpected due to their severity. So the RSA is the reference safety information, and it is a list of all medical events which defines which reaction are expected for the investigational medical product. So it may be contained in the summary of the medical product characteristic or in the investigator brochure. So the assessment of the expectedness includes a reaction is only considered expected if it is listed in the RSA and at the same specificity and severity. Also, a reaction shouldn't be considered expected because of the patient's medical condition, but rather depending on the investigational medical product. And for the safety reporting, this is for the drug that is used within the license and should include a summary of the medicinal product characteristic, describing the properties and the condition of use, where if the drug was used outside the license, this will depend on the risk, whether an investigational medical product or SMPs, and should describe the physical and chemical features of the medication and may be produced by pharma company or developing of the drug, and therefore should be included in details. So an investigational brochure will usually contain multiple lists of adverse reactions in different sections or across the document or in the appendix as well. So the section which should be used as the RSA must be identified by a clear title as a reference to safety information for assessment of expectedness of a serious adverse reaction, or ADRs. In this appendix are the reference for the expectedness used by the regulatory authorities. So only it should contain the SARs that are also to be listed in the investigational brochure in the section and should be in a table using the current MDR terms of the International Medical Dictionary for the regulatory authorities. So these can be found online at certain medical websites and should include a list of undesirable side effects, all adverse reactions, as well as it may have information on the population, whether they are elderly or children, as well as is specific to a product that may vary in the formulation, whether injected or oral, and should give frequency of the expectedness of the event. So they should follow up on all adverse events, depending on the factors, as well as until the resolution of the event is stable in all cases. So we should record or stop the date and the outcome of these adverse reactions. And for the serious adverse event, we should stop the date event seized by the serious or any later information that is recorded. So the report usually on other copies of this AE from the amendment added in these cases. Also, an important consideration when we are developing investigational medical product trial is blinding the study. And unblinding is also as important to be assessed or developed and following the protocol for. So the blind should usually be broken for a serious or unexpected adverse reaction, and the unblinded individual is subject only, and we should unblind as few people as possible within the same team. Therefore, the process should be covered in the protocol or in the NSOB. So the oversight of adverse event should be in the commercial trial the sponsor responsibility, and usually should provide reviews and oversight. Also, the academic sponsor is the Data and Safety Monitoring Committee responsibility, and they should review the new SAEs and SARs on regular basis, for example, weekly, and they should review any new SARs immediately within 24 hours. So oversight will depend on the Data Safety Monitoring Committee that should review all serious unexpected events, perform an trend analysis, review the reporting procedures on regular basis, and they must include independent members or experts. So it must include the independent member experts and should propose trials clinical for these reports. Also, in the case of developing a serious breach, it is defined as a breach which is likely to affect to a significant degree the safety of the or the physical or mental integrity of the subject of the trial, or even the scientific value of the trial. So the Medicine for Human Use Clinical Trial Amendment Regulation in 2006 regarding these factors is developed. So the safety breach can include breach of the GCP or the protocol leading to death, also hospitalization or permanent disability with eligibility violations and investigational medical product dosing error, or even failure to report these effects with urgent safety measure that is not implemented at all sites, including as well fraud or persistent use of wrong patient info sheet with systematic error in data recording or entry processing. So this might be identified by the monitorer or the auditor, also by the participant themselves, the researcher or investigator. So the report should be sent out to the sponsor immediately, and the sponsor must inform the responsible authorities within 7 days of becoming aware of this error. And the urgent safety measure can be implemented immediately without waiting for an amendment to be approved. And the informing of the required authority should be done by the phone, but should be followed up by writing within 3 days. We should inform them about what measures have been taken, the reason on why these measures were taken, and the amendment form or the covering letter as well. So in case of temporary halt of the trial, we should inform the responsible authority within 15 days, and having substantial amendment from a form to stop the trial, and having another substantial amendment form when we want to restart the trial, or in the study notification form. So annual reports include the annual progress report and the developmental safety update reports. And at the end of the trial, we send an end of study notification with final report within 12 months. Also, an annual progress report should be submitted to the required ethical authority at the anniversary of the original ethical approval in this case. And we should update the required ethical committees based on these factors. Also, the Developmental Safety Update Report was introduced in 2011 to improve the harmonization and was sent out to the required authority on an annual basis. And these can be found on the GSCO website with safety reporting and serious breach recommendations. So what should we include in the DSUR? In the executive summary and the main report should include introduction, word count, and marketing approval status. Also, action taken into the reporting period for the safety response, as well as changes to the RSI, inventory of the clinical trial that are ongoing, estimating the cumulative exposure, data in line of listing and summary stabilization, as well as any significant findings from the clinical trial during the reporting period, and any relevant findings from the non-interventional studies, as well as other clinical trial study safety information and safety findings and literature review or lack of efficacy if it was found in these studies. So all of these methods can be sent out depending upon our requirement for the required authorities. We will be taking a break now for Salat al-Maghrib and then continue our discussion. Everyone. So as we have discussed, the quality assurance and quality control, an important aspect to ensure the quality and the integrity of the research is to have an informed consent. So who does the informed consent affect? So this affects those participating in the research in the health and social care setting, as well as those who's hosting the research at their establishment, and those who fund the research proposal or even fund the infrastructure where the research is carried out, and those who manage the research activities, and those who are actually doing the research, meaning the research team. So what is the main purpose of the ethical review of the research? Is to protect the right, the safety, and the well-being of the research participants. So this is the most important part when we are conducting the research. Therefore, an informed consent has been developed. So ethical principles of research are to include the key philosophical principles that underpin our ethical values, including the four core principles of beneficence, non-maleficence, and justice with autonomy when we are carrying out the research project. So the first one, the principle of respect for the individual autonomy, is that individuals must always be respected, independent of their moral agent, with the right to choose how to live their own life and how to make their own decisions. The second, the principle of beneficence, is that one should strive to do good where possible and to do no harm. And non-maleficence focus on that one should avoid doing harm to others whenever possible. And the principle of justice is that people should be treated fairly, even though that this doesn't necessarily mean being treated equally. So according to researchers, they stated that they don't rank these principles in a priority order, but rather many argues that autonomy has become the dominant principles in these factors. So all of these are equally important, with no one prevailing over the other, but all should be included in the research process. So it is important to remember that not everyone agree with these principles, and many argue that they act simply as a checklist, and that they are shortcut to really assessing the morality of the issues. However, they do need to become the key concept which the health specialties work for, and certainly the basics for ethical concepts. So these include having, as we mentioned, as an informed consent at all cases, also having honesty and integrity, confidentiality, as well as anonymity when we are recruiting patients, or especially in focus group when we are interviewing people with certain sensitive information. Also valuing the diversity within the society by including vulnerable groups in most research projects, and scientific validity and competency whenever possible. So beneficence is the active promotion of the welfare of others. We're doing good for others and not harming them. Also, to do good or to intend to do good as well is to improve, for example, diagnostic procedures, uh, therapeutic or prophylactic procedure, and promote understanding of the etiology as well as the pathogenesis of diseases. So all of these can be included as the beneficence for both the society as well as the patient and patients with the same condition. So these can include actions protecting the right of others, preventing harm, helping those with disability, and rescuing those in danger. So in the hospital setting, this can frequently clash with the respect for autonomy, which is the paternalist that doctors would know best on what to do to the patient and how to treat them. So benefit can be, as we mentioned, benefit to the participant themselves, benefit for the patient with the same condition as well, also benefit for the medical and nursing staff by improving health of the patients, as well as benefit for the health system, benefit for the researcher in themselves, and benefit for the society in general in terms of improving the population health. So non-maleficence is essentially the requirement to do no harm to others in most cases, but is not the same as being wronged. And these can include don't kill, don't harm, don't cause injury or pain, as well as do not incapacitate and do not cause any offense. So these can be used to support issues of confidentialities, but we need to think about the hospital setting and these steps to be included, as they can, in many instances, contradict this sentence. So some can as well overlap with beneficence in some cases, and therefore can be indicated in some of these instances. So what is harm? This can include physical harm, such as side effects of the investigational medical product, or even withholding a beneficial standard treatment so that the patient is worse off through taking part in this research project. So we shouldn't harm the participant at any cost. Also, the psychological harm, this can include fear of the unknown or fear of being sick, also corrosion to choose to participate in the study, For example, when the patient know that there is no treatment for this condition, therefore they are more likely to be coerced into participating in the trial. So informed consent is defined as a process by which a subject voluntarily confirms their willingness to participate in the trial after having been informed of all aspects of the trial that are relevant to the subject's decision to participate. So informed consent in clinical trial research is documented by means of a writing of a signed and dated informed consent form in all of these cases. So autonomy is regarded as a self-rule in which a person should be free to make their own choices and decision free from pressure or even interference from others to take part in the trial, provided that they have mental capacity to make their own choices and the information required to enable them to make these decisions. So the respective autonomy cover issues such as consent, choice of the treatment regimen, the decision to refuse a life-saving therapy, also willingness to take part in the trial with other research healthcare settings, as well as treatment of children and those with limited mental capacity who are unable to make their own choices regarding their treatment. So respect for autonomy is the power or the right of self-governance in which a manpower is possessed for a reason to give a law to themselves. So a person who is autonomous is capable of making their own choices and can make their own decisions. Also, to be encouraged, not just passively allowed to make their own choices, is the main concern in the respect for autonomy. Also, non-autonomous groups or those with limited autonomy are said to be vulnerable groups. So vulnerable groups will be discussed later in details to understand who are regarded to be as a vulnerable person. So for an informed consent to be valid, it shouldn't just be informed, but rather the full information should be given to the participant. Also, it should be clear information that is understood with no corrosive, and the information should be recorded for the participant to take with them, as well as continuing updating the participant on their trial participation and any associated funding. So for the informed consent to be conducted with integrity, this should be fully informed, with no deception and no corrosion, with no inducement giving them money to participate in the trial, but rather they should be volunteers. So they should also be free to withdraw from the trial at any time without having to give a reason or a care of being negatively affected. And this should be explained clearly to all participants when or before they take part in the study, is that they are free to withdraw from the study at any given time. So, vulnerability is regarded for certain individuals, for example, those with cognitive vulnerability, such as minors, elderly, and people with mental problems. Also, medical vulnerability is regarded for people who are severely ill or in emergency care where no cure is available. Also, there is the institutional vulnerability. This can include, for example, prisoners, hospital inpatients, military personnel, or children in school. Also, the differential vulnerability. This is due to dependency and participants being on patients with subordinate staff, students, or any other who is in a lower hierarchy. So, family or social hierarchy as well can be regarded as social vulnerability. Also, there is the economic vulnerability. These are in the low-income groups, such as students, elderly, and anyone with low income. So, social vulnerability as well. These are people with mental or behavioral problems or ethnic minorities, especially due to language problems. Also, there is the situational vulnerability that is due to current job, family state, and etc. There is also the emergency situation and those who are often time limited to make their own decision in a critical condition. Also, for justice, in healthcare, this is largely related to access to medical provision or insurance where required. So, justice would seem to require fairness and equality, but healthcare provision is cost restricted in some cases, and therefore medical resources should be distributed based on some sort of adjustment. So some of the approaches to ethical principles included the utilitarianism, which is a goal-based approach. So if the intended outcome is worthwhile, then the mean to achieving it is justified in this case. Also, the burden on the individual is justified by the overall gain to the society as a whole, and it allows the rules to be broken if the total good will outweigh the harm for those who follow this principle, but as such, it shouldn't be applied in clinical research. So the issues with this approach is that it can add up good and add up harm. Consent is not as essential with this approach. It will resist the right of the individuals to drop out of the research and could continue with the trial to the end of the even if the group is clearly being disadvantaged and allow for euthanasia. So this approach is ethically unacceptable and shouldn't be followed. There is also the duty-based approach. This is a moralistic approach in which breaking the rules is considered to be wrong. Also, the individual is all important, and duty of care to the individual participant will outweigh the welfare of others, the researcher, or the society in general. Also, for the duty-based problems, these are when the laws, the moral codes, or ethical codes conflict, such as when telling the truth will cause psychological harm. Also, when there is no relevant law that exists, as well as differences in what is permissible between the countries, and when breaking the duty to the individual might prevent serious harm to others as well. Also, there is the right-based approach in which the individual needs will outweigh the needs of these participants. in most cases. Also, it will allow rules to be broken if the rules violate the person's right, and respect for the individual autonomy will override causing them harm, and it will have civil rights, freedom, and human rights as well. So in the rights-based approach, this will allow enthusiasm on the quest, as well as allowing people to volunteer to be harmed. Also, it is a human rights act that is adopted in the UK in most cases. So the NHS ethics protocols draw on all of these approaches. By defining the rules and the codes, it becomes predominant duty-based approach. So why do we need a formal ethical review process? This is independent external viewpoints to identify any unintentional problems, such as corrosion, burden to the participant, bias, and scientific validity, and including the public confidence. So for risk assessment, we should assess for hazard with features that has the potential to cause harm, such as well as risk that have the likelihood to be hazard, which will actually cause harm. Also, consequence, which is the outcome of the harm if it should occur, and consequence risk analysis. So, what can be risk assessed? This can include the processes such as venipuncture, the equipment that such as electronics, the patient as well for assessing for risk falls or leg ulcers, for example, the medications or the facility, as well as the study protocol or the ethics in general. Also, this can include assessing the drug history and the preclinical work, as well as the dose, the starting and escalating, the controls, the communication, the pharmacovigilance, and the procedures that are to be included. So for the risk minimization, this can include, for example, making a tea in which in boiling water, we should limit the quantity to avoid overslip. In the electric supply, this can include the safety check. Also, the allergy to milk supply, this can include the participant awareness. The allergy to milk, the sugar, for example, include the participant awareness if they are diabetic. The spillage or breaking up, this can include the safety check and the training at each step. So in summary, historic events have shaped the regulation of research involving human subjects. This has led to the introduction of GCP regulations and governance procedure for research. Also, research ethics, GCP, and policies framework for health and social care research are in place to ensure the protection of the right, safety, and well-being of research participants. So as we have discussed the design of qualitative research, we will continue with discussing the quantitative research study design and methods. So QD is based on the scientific method, quality trial design. Also, it used deductive reasoning, and researcher forms a hypothesis, the collection of the data and an investigation of the problem, and then they can use the data from the investigations after the analysis is made and conclusions are shared to prove the hypotheses that are true or even false. So there are four types of quantitative design. These include experimental studies, which are comparative correlation studies, and surveys. And for the experimental studies, these often are called true experimentations and often uses the scientific method that are established with the cause-effect assessment to assess the relationships among the variables in a research study. Also, researchers can make an effort to control for all variables except the ones that are being manipulated, which are the independent variables, and the effect of the independent variables on the dependent ones that are collected and analyzed for a relationship. So even though that questions may be posed in the form of research or experimental research is guided specifically by a hypothesis. So sometimes experimental research can have several hypotheses, and a hypothesis is defined as a statement to be proven or disproven for its reliability. So once the statement is made, then the experiment should be started. So the major feature that distinguishes experimental research from other types of research is that the researcher can manipulate the independent variables. So subjects are randomly assigned to experimental treatment in this case based on sampling methods. So experimental design should be developed to ensure the internal and external validity of the study. So the internal validity are the result of the study caused by the factors included in the study and that they are caused by other factors, exterior variables, which are not part of the study that were examined. So external variables are unwanted variables that may have an effect on the experimental outcome. For example, if we are worried about gender, then we should only include one gender in the study to assess for the actual impact. Also, there are some threats to the internal validity of the study. These can include the subject's characteristic, which are known as selection bias. So the groups that may have been different from the start, if we were testing the instructional strategies to improve reading and one group enjoyed reading more than the other group, then they may have improved their reading because they enjoy it rather than the instructional strategies that they were using. Also, location. Perhaps one group was at a disadvantage because of their location. And examples can include, for example, the cities that have been demolishing a building next to one of the school in which we are studying. These can result in obstruction of the participant commute to the trial center. Also, there is the instrumental decay, in which the testing instrument may not be scored similarly. Perhaps the person grading the post-test is fatigued and can pay less attention to the last set of papers reviewed. So it may be that these papers are from one of the groups that will receive different scores from the earlier group papers. So these will depend upon the instrument decay that was used. Also, there is the data collection bias or the collector bias. These include that the person who collects the data may have favors one group or some characteristic with some subject that possess over another. Example include a teacher who favor a strict classroom management that may rate student attention under different teaching conditions with a bias toward one of the teaching conditions. There is also the resentful demoralization of the control group, in which the control group may become discouraged because it is not receiving the special attention that is given to the treatment group, and they may even perform lower than the usual because of this factor. So once the researchers are confident that the outcome of the deviant express of the experiment they are designing is the result of their treatment, then an internal validity, they can determine which people or situations with the result of their study apply to the external validity. So the external validity are the results of the study generalizability that can be applied to other populations and settings. These usually come in two forms, which are the population validity and the ecological validity. Also, there is the population validity, which is the extent to which the result of the study can be generalized from a specific sample that was studied to the larger group of subjects. Where in the ecological validity, these are the extent to which the result of an experiment can be generalized from the set of environmental conditions created by the researcher to other environmental conditions or settings and conditions as well. So the threats to the external validity include the pretest sensitization, in which a treatment might only work if a pretest is given. Because they have taken a pretest, then the subject may be more sensitive to the treatment itself. So had they not taken the pretest, then the treatment wouldn't have worked in this case. Also, the measure of the deviant validities, in which a treatment may only be evident with certain types of measurements, such as a teaching method may produce superior results when its effectiveness is tested with an essay test. But it can show no difference in the effectiveness when it is measured with an MCQ test, for example. So first and foremost, an experimental study must have an internal validity. And if the researcher cannot be certain that the result of the study or the experiment are dependent on the treatment, then it doesn't matter to which people or situation they wish to generalize or apply their findings. So the important external validity is reliant on having an internal validity in much the same way that the validity of a measurement instrument is reliant on the instrument that is being relied. So some of the different research designs include one-shot case study design, also the one group pre-test post-test design, the statistic group pre-test post-test design, and statistic conversion design with randomized post-test only with control group design. There is also the randomized pre-test post-test control group design and randomized Solomon 4 group design. So all of these are different methods or designs that we can use when we are conducting quantitative studies. So to understand how a drug behaves in vivo specifically, we need to capture the pharmacokinetics of the data to assess for toxicity and assess for efficacy. So measuring the exposure at different doses will assess the absorption, the distribution, metabolism, and excretion. So defining the tolerability and the safety of the maximum tolerated dose will assess the well-being and the tissue damage of these factors as well. So this is an example of a duration of repeated dose toxicity studies to support the phase 1 and 2 trials in Euro and phase 1, 2, and 3 in the US and Japan. So the duration of the clinical trial for a single dose should be at least 2 weeks, where if it was for up to 2 weeks in more than a single dose, and if the dose was to be repeated for over 6 times, then it should be for over 6 months. So the repeated dose preclinical studies commonly have 3 dose levels and a vehicle with a low dose that should be non-toxic, but small multiple destination therapeutic doses to be included. So high dose should be sufficient to cause toxicity, but not death, and an intermediate dose should be therefore applied. Also, detailed observation of the physical condition, growth, behavior, and biochemistry, as well as any other findings, should be assessed at all of these phases. So some of the common terminology that are discussed in research design are the NOEL, which is the no observable adverse effect level. There is also the HED, which is the human equivalent dose, as well as the MABL, which is the minimum anticipated biological effect level, and the MRSD, which is the maximum recommended starting dose. So the NOEL is the highest dose level that doesn't produce a significant increase in adverse effects in comparison to the control group. So this refers to the biological significance rather than the statistically significant. For example, it doesn't have to be statistically significant. Also, it distinguishes from no observed effect level to that it may affect in animals that are acceptable and lower observed adverse effect levels than in humans. So there are essentially three types of findings in the non-clinical toxicology studies that can be used to determine the NOEL, including the overtoxicity, in which, for example, clinical signs, macro and microscopic lesions are being observed. Also, the surrogate marker of the toxicity, such as the serum liver enzyme level and the extragated pharmacodynamic effect that can develop with certain conditions. So the human equivalent dose after the NOEL in the relevant animal studies have been determined, then they are converted to the human equivalent dose to assess for their absorption in humans. So the assumption of the HED is that the toxic endpoint for the therapeutic administered that are systematically to the animals, such as MTDs, are usually assumed to scale well between species when the doses are normalized with the body surface in general. So, for example, for anti-epileptic drugs, then the doses that are lethal to 10% of the rodents and the MTDs in the non-rodent both correlate with the human MTD when the doses were normalized to the same administration schedule and expressed as the human equivalent dose. So HED is calculated from the NOEL in the most appropriate animal species uh, using a body surface area conversion factor. So the most appropriate species is usually the most sensitive one, but occasionally for biologic treatment, another model is more relevant, for example, in genetically manipulated animals. So this is an example of how to convert an animal dose to a human equivalent dose based on the body surface area. So when we are converting the human equivalent dose, the first step is to convert the animal from milligram kg to milligram per meter square. So the dose in the mg kg is multiplied by the conversion factor indicated as kilometer for the mass confessed. So Km factors has a unit of kg per meter square that is equal to the body weight in kg divided by the surface area in meter square. So this is the formula that we should follow and an example of how to convert a dose. So the dose can then be converted back to mg per kg in humans by dividing the dose by the Km factor in human to ensure for adequate sufficient conversion factors. So the HED can be calculated directly from the animal dose by dividing the animal dose by the ratio of the human animal factor as well. And in this table, it can show multiplying by the ratio of the animal to human factors with the fourth column that is to be shown. So after that, we should apply a safety factor. This includes that to be applied to the HED to increase the assurance that the first dose in the human will not cause any adverse effect as a result of human bioavailability and metabolism that may differ significantly from that of animals. Also, the mechanism of the toxicity that is may not be known, such as toxic accumulation in the peripheral compartment of the body tissues. Also, the toxicity that may be due to unified metabolites that are not from the parent drug as well, and toxicity that may be difficult to detect in animals, such as myalgia, headache, or ocular problems. So the MRSD, in general, one should consider using a safety factor of at least 10 when we are converting the human dose of the chemical product. So dividing the HED by a safety factor will give the MRSD, but the safety factor may be increased in cases of inadequate dose response data, also due to novel therapeutic targets or even to animal models with limited utility, as well as steep dose response curve and severe toxicities or non-vulnerability of the toxicity, as well as toxicity without predominant signs. Also, irreversible toxicity or unexplained mortality and large variability in the dose or the plasma drug levels, eliciting their effect. Also, these can be non-linear pharmacokinetic impacts that are to be assessed. So, in the efficacy studies, we assess if there is any signals of desired impact on the participant. Also, it relies on the animal models of the disease of interest. And in animal study, most of them fail to predict the efficacy in humans. This is mostly due to that animals are not miniature humans. Therefore, differences in metabolic pathways and drug metabolism will lead to variation in the efficacy as well as in the toxicity level. Also, inadequate knowledge of the cause of the disease mean it is difficult to reproduce the human conditions, and differences in the route and dose of administration compared to human will also be present. In addition to this, lack of a robust design with methods of randomization, blinding, and choice of conversion therapy can therefore be contacted as issues to fail predicting efficacy. Also, a small experimental group with inadequate statistical power and simple statistical analysis that doesn't account for confounding and failure to follow the intention to treat principles will lead to failure to predict the efficacy, as well as using outcome measures of uncertain relevance to the human clinical conditions, and that animals don't report the subjective effect and variable duration of follow-up, which may not correspond to the disease latency in humans. Also, it may need to humanize the animal for some treatment to be conducted. So these are issues related to the reproducibility relating to the condition of the medication and the reported reproducibility concerns. And these include the review of the study design. So, improving the bioscience research reporting, the arrived guidelines for reporting the animal research has been developed. So these include the housing, the sample size, allocation of experimental group, and the experimental outcome in general. These as well include the baseline data to be collected, the number of analyses, and outcome estimation in most of these cases. So some of the alternatives to using animals in the treatment include induced blueprint of the stem cells, creating disease on an isogenic background, or organs on a chip by combining cell types or on a scaffold, as well as in silico studies with computer models of the disease and network medicine, as well as the human as an experimental subject in some cases with microdosing, the pharmacological challenges, as well as imaging procedures.
Assalamu alaikum wa rahmatullah. After understanding the design of studies, the quality control, and the assurance, we need to understand the plagiarism and paraphrasing procedures. So the first step is the literature review. And why do we need to review the literature? These include to support the argument for the need of the research. In other words, to give reason why the topic is so sufficient, important for it to be researched. Also to provide the reader with a brief up-to-date account, discussion, and on issues that are relevant to that topic. Also to discuss the relevant research that is carried out on the same topic or similar topic, and to prevent the repetition and duplication. This will also be needed to support or define the statement of the problem, to determine the suitable type of methodology, and outline the problem or topic and the need for the research in this particular area. This will also be used to appropriate recent literature review about other work in the field. So why do we review the literature? This is to start in a systematic way by defining the topic, the cause of the study, and why there is a need to study this topic. Also, the source that is referred to that should be relevant to the problem, and the literature review that include a range of written sources and studies included that should only actually read by the researcher. So the sources of information can include the community, the district or province level, including clinic, hospital, or local survey, for example. And at the national level, this can include articles from national book, previous researcher in the university library, or the Ministry of Health as well. And at the international level, this can include information from the WHO, for example. So any source of information that could be from individual group or organization, and information could be from a published source, such as book or magazine, as well as that the source could be from an unpublished information, such as reports or previous unpublished pieces. And this is an example of how to write a source or reference a source using a Vancouver style. And this will depend upon the source of the information that we have used, whether it was a book or a website. So each one will have a different format. The other second common referencing method is the Harvard style, and most articles or magazines will only require Harvard or Vancouver style. And if we are including a reference from a source of the internet, we should always include the link of that website and the date in which we have accessed that information. So the ethical consideration include in terms of the authors, the name of them should be stated at the end of each statement, even if it was a verbal or personal communication. They should also review the studies which stand with or against the study problem, and not only the one of them. So this will avoid any bias, which is the deviation of result or inference from the truth or the process that lead to such deviation. So bias is also any trend in the collection, interpretation, or review of the data that can lead to the conclusions that are systematically different from the truth. So bias is also the distortion of the available information in such a way that it reflects the opinion or conclusions that are not representative of the real situation. So people should consider the first ethical permission before carrying out any research. This is usually obtained from the local area in which the study will be carried out, such as hospital or ministry or university. There is also consent form in which the population under study signs or gives verbal approval to that they agree to take a part in the study and that they have the right to withdraw from the study at any time without having to force them or give information that are provided by them that will be treated on the privacy and confidentiality. So for the reference management, when we are writing a research, we should use a reference management program such as EndNote and Covidence, which are the most common ones. So the learning outcome of using reference management are to recognize why reference management are important and to understand how they will help in our studies. Also to be able to find access in EndNote and to import reference from a variety of database and websites into EndNote, as well as to add in-text citation to the word with EndNote generate reference list and to understand further features of EndNote and where to access support and guide. Also to be aware of Covidence and how it can help with the literature review and how Covidence interacts with complement reference managers. So first of all, what is plagiarism? It is the representation of another person's thoughts, words, or images and diagrams as though they are our own. So To comply with a good academic practice, we need to quote or paraphrase to avoid using plagiarism or being caught in plagiarism. This can be conducted by citation or having a reference list. So citation is when we use the same words of the participant. Therefore, we can put them in citation or quotation points to state that this is a citation. Where in the reference list, we write a reference when we use information from a certain website, but we don't write the same way, but rather in another way that based on our understanding. So quoting, we use the exact words of an author in our assignment, but we use quotation marks that will help to support our argument and help to reinforce or raise a new point. So this doesn't mean we can just cut and paste large section of text, insert quotation mark, and hand in our work, but rather we should only use citation when the information is very important to include and cannot be written in another word. Where for paraphrasing, we should read a piece of work and then rewrite it in our own words by interpreting the ideas of the original text for our readers. So paraphrasing a document that we have understood the academic context of the piece and allow us to support our argument. So it is not word switching, but rather writing what we understood and what we have found to be related to the research we are carrying out. So citing is when we use another person's work, either by referring to their ID or by including a direct quotation that we must acknowledge this in the text of our work. Where the reference list is in a list at the end of our work or our research paper, where we should include a reference to all the sources of information that we have used, and this is called our reference list. Also for bibliography, there may be an item which we have consulted for in our work but not cited, and these can be listed at the end of our research paper in a bibliography chapter to ensure that we give each person the credit they deserve and who has helped us in our research project. So when we are citing in a Vancouver style, each piece of work that we have cited in our text should have a unique number that is assigned in the order of a citation, beginning from 1 until the end of our research paper. So if we have cited a piece of work more than once, then the same citation number should be used for that work. This is an example with a Vancouver referencing style. As you can see, we have used numbers and Vancouver style of 1 and 2. And then at the end of our research paper, we write a reference list and include those references in the order they appear in the text. So in Vancouver style, we list all the sources that have been cited in the text of our work. The sources in our reference list should be in a numerical order. Each number matches the reference to the citation in the main body of our text. So using reference management software will help us from having to rewrite all reference at the end or having duplications of the reference. First of all, we need to download the reference from the online database, create our own library of relevant references that we have used when reviewing the literature. This will help us to store the PDF with the reference so we can return to them at any point. Also, we need to identify and delete the duplicate. The program will help us to do that. It will help us to organize, sort, and search our reference library into projects and sections. It will help us to share the library with a group or the research group. It will help in inserting correctly formatted citation from reference work library into word processing documents. So it will also generate reference lists in a variable output styles, including Harvard and Vancouver styles. So EndNote is the most commonly used referencing program. So the first step in using EndNote is to set up an EndNote library after we have downloaded the program. We will first create and save our EndNote library, preferably to name it with the same name as the research, so it will be easier to follow. Then we can import the journal terms list by adding them one by one and searching for them. For example, you can search for a journal by the title, Journal of Allergy, for example, and then we search in that journal for the term list. So these can include preset lists of common journal titles divided by the subject, and for each title, there is a list of common abbreviations, and the term lists aren't exhaustive, therefore we can use at each step. So the second step after we have created our list, we will import the PDF directly into EndNote. After that, we will import the citation information from the database and the website into EndNote. And the third step is to insert the citation into the document and generate a reference list after that. So the feature that EndNote has is that it can automatically go and find full text for us. It can import the PDF from our EndNote library. It will help us in searching for and adding citation information within EndNote in itself. It will help in sharing the library with our group or research team as well to highlight, for example, any important information to be included. Other options other than reference or EndNote is to use reference work, to use the Z-Through, which supports sheets that are available on the library website. Also, Mendeley, which supports the sheets that are available on any library website. Using reference management software will help us to match a product that we need based on our conversion and needs of the research project. There is also confidence that we can use to help us with reference management. This is a collaborative online tool for better systematic review management. So confidence is important in systematic reviews. This will help us to have a faster review with an average of 35% reduction in the time that was spent per review. There is also the data security in one place, as we will never lose track of our review and the data that are stored safely and centrally within the program. Another advantage is that it has seamless collaboration that enables the whole review team to collaborate from anywhere and to carry on after their own work. It can also suit all level of experience that are easy to use for both students who are new to review and also for experienced researchers. So, how does Covidence fit into the systematic literature review process? We will first search for multiple databases, and we may also do so in other searching engines, such as looking for journal table of the content list of the key journal, or even by looking at a reference list. Second is to use the RSI of the PubMed, for example, or EndNote files. And third, it will auto detect any duplications from the citation and will delete them. And in Confidence, we can choose yes or no, or even maybe to decide if the article is suitable for further screening. And in Confidence, we can choose to include or exclude to decide which article we will include in our review. Also, we can use the export option to export the citation information in our paper reference management. And it can use the data extraction for risk of bias tools and Providence and to export any flowchart data from Providence to put in our own research paper. We will be taking a break now for Salat al-Isha and things.
السلام عليكم ورحمة الله وبركاته. معاكم مها القحطاني Clinical Research in King Abdullah International Medical Research Center. اليوم إن شاء الله بنتكلم عن موضوع استكمال للمحاضرات Research Ethics. The aims of today for the lecture is to increase the awareness of research ethics principle, to encourage a connection between your research projects and internationally recognized principle of research ethics, to offer guidelines of on school and university research ethics procedures. What is the research ethics? Is a worldwide set of principles governing the way any research involving interaction between the researcher and other our human tissue, our data relating to humans is designed, managed, and connected in preparing a research project. The dignity, rights, and safety and well-being of human participants must at all times be considered, respected, and safeguarded. فالresearch ethics, I will talking Arabic and English. الريسيرش ethics هي مبادئ لابد أن تتخذ في الأبحاث لحماية المشارك في البحث. All projects conducted by university staff. دقايق بس معلش. All projects conducted by university staff or students that involve a human participant in a way that might harm or absent them. However, slight the possibility or where they can be deemed to be in vulnerable or disadvantaged situation must receive approval from a recognized research ethics committee اللي هي اليو ار اي سي اللي راح نتعرف عليها في ال front of the lectures. An ethics committee will assess whether we know the research ethics committee are responsible for review ethics applications in order to ensure the indicate consideration has been given to ethic aspect of research project, reduce the potential for harm and absent to the participants. So the research is justified, like example, is the research likely to add to the existing knowledge base, and it is of sufficiency standard, including whether the research are qualified to carry out the rules proposed, the risk it poses to participants outweighed by the potential benefit of the research. So the research appears to comply with all statutory and other guidelines. Data management and the handling appears to comply with the relevant legislations, and the financial arrangement appear sound. So it's very important for the ethics committee to assess this in the process and research. There is principle of research ethics are grounded under these hindings. So this is hind point under the process of the ethics research is autonomy and beneficence and non-maleficence and confidentiality and integrity. إنه يكون فيه نزاهة في البحث وسرية في البحث، وأيضاً إنه يكون فيه الأوتونومي والسرية في the process of your research. We know in all research proposals are to security to ensure that these principles are addressed in practice. So what's this meaning? That means the researcher report and review security response to the questions and the process can seem draw and out and complex, but the aspect of research ethics and essential simple. So in practice, what is the research? You should, you know, what's the process in your research, what is the research and why it is important, who is involved in your research, what are you asking people to do, what are the ethic issue involved, and how will you manage this? So what does this mean in practice? We needed to think about whether the research is necessary, identify the specific risk it poses you are present or represent for your research participant, including develop the plan for meaning those risks or develop a concentrated approach to obtain informed consent. Be clear with participants about the level of confidentiality or anonymity on over. So the reminder, you should reminder of this session will be looking in details at the process of applying for the ethics approval for the protocol or proposal in your research. There is 4 steps in the process you should concentrate in your research in ethics process. Develop research proposal with supervisor if appropriate, and research questions rational, significant, and who is going to be involved and how ethic issue. 2, decided what level of review do I need, and 3, complete an application view the university online system and appropriate to the level of review required. ethical review manager, we will see this meaning أو terminology in front of the lecture. يكون في اللي هو for submit for review to supervisor, our school, our UREC dependent on level of risk. We work the principle, we know we should concentrate about we work to the principle of proportionate review, less security required for low risk projects and more for high risk projects. So the level of security applied must be proportional to the risk involved. Sometimes approval from supervisor is enough, or sometimes as application from ethical approval will need to be made to the University Ethics Committee. Committee is sitting each week in semester time, like the study for PhD or masters, for the process for approve for ethics research. Sometimes an application to the school research ethics committee will be needed non-deadline all year around الBGR and the MA courses. What level of review do I need? عندنا في 3 levels for the risk for your research and ethics. low risk أو medium risk أو high risk. متى يكون في low risk review by supervisor أو in the school, أو medium risk review by school research ethics committee. We should note that all staff applications are reviewed by the University Research Ethics Committee and regardless of level of risk. We will know about all what's the high risk. High risk, the research focus in group within the society in need is special support. Is carried out in unstable or violent setting, involve non-standard methodology. Our approach present risk to the personal safety of the research. Our research participant beyond what is normal in setting our where there is possibility the research might be become distressing to the research. Our research participant in 1 hour more away. High risk, we should concentrate application are reviewed by the University Research Ethics Committee after initial screening at school level. Medium risk, the research in young adult and children, like accredited settings such as culture institute, our school, our in a club, our combined by a career, our professional with a duty of career. Follow standard procedure and establish research methodology as carried out in public setting that does not present risk to the research. This is meaning of medium risk of research participant, and what is normal in the setting does not require research participant to provided personal and sensitive information, like to lead significant level of dangers or distresses. The research topics are either not continuous or sensitive, this meaning medium risk. Our response personal would agree the topic of legitimate or interest and may result in distress and rare. So medium risk application are review at school level. اللو risk يكون عن طريق اللي هو not required research participant to provide personal and sensitive information, like to compromise them or to lead to distresses, like the research topic are not continuous or sensitive, such asking about religion, belief, or social preference. So So such as asking about religion, beliefs, our social preference, and also the student project demand to be of low ethics risk required in the approval of the supervisor, or but still need to be submitted to ERM. عندنا is my research low or medium risk. We should ask these questions about your research when you start your research. Like low risk research typically does not focus in continuous or sensitive topics, does not involve collection of personal or sensitive data, focuses in professional practice or professional roles, takes place within normal working hours and in public setting, may involve the collection of photographs or videos or participant, but only where the research can guarantee control access to authorized viewing. So good research and ethic research, drawing the line between good research and ethic research is not always clear. Good research is concerned with integrity and soundness of data, whereas research ethics is concerned with the means the data was collected. Research ethics is concerned with well-being of other society or other people, animals, while standard concern. So issue in research ethics like conduct of individual, our research fraud, our mistreatment of lab animals, accuracy and honest in report result, plagiarism, violating of intelligent property rights, conflict of interests. Researcher among each other, our researcher and university, it's important to concentrate about this for conflict of interests between the sponsor, our university, or the center research. So the principle of the contribution to science as a whole, check of research harm full to the society, our individual, how university should resolve their conflict among themselves. عندنا اللي هو البيسك ethical principles, important the expression basic ethic principle refer to this general judgment that serve as a basic justification for the many particular ethic, like respect for personal, and the person should be treated as autonomous, and the person with diminished with autonomy is entitled to protections. Also can think and discuss his personal goals and act under those goals. So protect who cannot make their own self-determination, or they are not eligible for doing so. عندنا اللي هو beneficence, أيضا اللي هو not only respect their decision and protect them from harm, but also make effort to security their well-being. Is important to concentrate think of this in obligations and not charity. Do not harm for the personal or human, justified. Also, يكون في right be treated equally. Someone is denied benefit for the reason. The principle of fairness and distribution. عندنا اللي هو بعض الترمينولوجي it should be concentrate in the ethics research, like independent ethics committee اللي هي ال IEC، وعندنا research ethics committee اللي هي لجنة أخلاقيات البحوث، والethical review board اللي هي مجلس المراجعة الأخلاقية، وعندنا الانستيتيوت ريفيو بورد اللي هي مجلس المراجعة المؤسسية. هذه terminology it should be concentrate in your research، وتواجهونها في بداية الأبحاث، وأيضا في اللي هي أخلاقيات الأبحاث البحثية. عندنا في أصول تاريخية صدرت منها أخلاقيات البحث. during World War from 1939 1945 كان في German doctors طبقوا أبحاثهم experiment على اللي هي الفيتال بيرفورم. فكان في، طبعا أنا خليتها بالعربي وبالإنجليزي توضيحا لبعض الحضور. طبعا عندنا اللي هي كان في طبقوا أبحاثهم بدون ما يكون في consent informed موافقة من اللي هم الهيومن اللي داخلين في الأبحاث. فاختبروا أدوية وطبقوا وأجروا طرق العلاج عليهم. فكانت طبعا هذه اسمها محاكمات نورمريج، تعد يعني أشهر المحاكمات المعاصرة في الأبحاث. طبعا عقدت في 20 نوفمبر في 1949. طبعا كان في سجن 7 سنوات وإعدام 12 سنوات. فهذه ترجع لأهمية أخلاقيات البحث وإجراء البحوث على الإنسان أو على الحيوانات. طبعا عندنا وثائق أساسية لأخلاقيات البحوث الطبية. عندنا اللي رجعت أصول التاريخية لأخلاقيات الأبحاث. عندنا وثيقة نوربرج، وعندنا إعلان هلسنكي، وتقرير بلمونت، والقواعد الإرشادية الدولية لأخلاقيات البحوث الطبية الحيوية على الإنسان في سنة 1949. أول وثيقة اللي هي نوربرج اللي كانت في 1947. طبعا هي a set of research ethics principles for human experimentations set as a result of subsequent numbering trials. فكانت عقدت على 10 points. عندنا إعلان اللي هو هلسنكي اللي هي Declaration of Helsinki. طبعا هو كانت a set of ethical principles regarding human experimentation developed for medical community by World Medical Associations. طبعا هي كانت على اللي هي المعذرة، شكل كان في مشكلة في الإنترنت، شبكة الإنترنت. إذا ممكن بس تعملون هوست لأنه ما قدرت أعمل شير. عندنا طب استكمالا، المعذرة كان فيه مشكلة في شبكة الإنترنت. عندنا ال Council of International Organization of Medical Sciences، اللي هي ال International Non-Government أو Non-Profit Organization، اللي هي ال World Health Organizations. عندنا طبعا هي كانت guidelines لعدة ل21 point، اللي هي كان ال Ethical Justification أو Scientific Validity of Biomedical Research involve a human subject، وأيضا كانت ال Ethical Review Committee، وعندنا ال Ethical Review of External Sponsor Research، وعندنا Individual Informed Consent وال Obtaining Informed Consent Essential Information for Prospective Research Subjective. عندنا سادس point اللي هو Obtaining Informed Consent Obligations of Sponsor and Investigators، وعندنا اللي هي Endorsement to Participant in Research وال Benefit and the Risk of Study Participations، وعندنا اللي هو Special Limitation Risk when research involve individuals who are not capable of giving informed consent. عندنا اللي هو Research in Population and the Committees with Limited Resources. اللي هو البوينت الآخر اللي هو Shows of Control in Clinical Trial، اللي هو Actable to Distribution of Burden and the Benefit in Selection of a Group of Subject and Research. أيضا اللي هي Research Involve اللي هي ال Vulnerable Personnel، اللي هي Integrity أو النزاهة البحثية في ال Ethics Research. عندنا الريسيرش تضمنت أيضا الريسيرش involving children، وأيضا الريسيرش involve individual who by reason of mental or behavioral disorder are not capable of giving educating informed consent. عندنا الwomen أيضا تضمنت أخلاقيات الwomen as research subjects، والpregnant women as research subjects. أيضا اللي هي safeguarding اللي هي السرية للبحثية في الأخلاقيات البحث. عندنا تضمنت اللي هي right of injury subject to treatment and compensations. أيضا عندنا strengthening capacity for ethic and scientific review and biomedical research. تضمنت أخلاقيات أيضا ال ethic obligation of external sponsor to provide healthcare service في ال CIOMS guidelines. طبعا عندنا ال ethic concentration for scientific research اللي لازم احنا ناخذها بالاعتبار ومهمة جدا في اللي هي الأبحاث السريرية أو الأبحاث النظرية، اللي هي المصداقية، truthfulness، وأيضا الإكسبرتايز، الخبرة مهم جدا، السيفتي، التروست، الثقة، إنه يكون في consent الموافقة من قبل المريض، إذا كان في withdrawal انسحاب من قبل المرضى أو قبل المسجلين في البحث. عندنا اللي هي digital recording، اللي هي التسجيل الرقمي، مهم جدا إنه أي document إنه يكون فيه تسجيل رقمي لحفظ سرية المعلومات البيشنت. وعندنا اللي هي الفيدباك، التغذية الراجعة. وعندنا الفولس هوب، إنه يكون في الأمل المزيف الكاذب مع البيشنت أو registration humans. عندنا اللي هو الاستغلال المواقف، اللي هي exploitations. وعندنا الانونيميتي، سرية المعلومات، والانيمال رايتس. طبعا هذه هي الethic consideration for scientific research. راح نتعرف على كل نقطة لأنها تعتبر من الاعتبارات المهمة في أخلاقيات البحث، ممكن يتجاهل عنها الباحث وما تسبب على interruption أو stop for your research. عندنا اللي هي المصداقية، يعني must be movable search result honestly and be honest with born and not be supplement any missing information or incomplete depend on what you think has happened. إنه تكون لا تحاول إنه أنت تدخل أي معلومات عن طريق اعتمادا على نظرية غير معتمدة أو عن طريق رأي آخرين. فهذه تعتبر من مصداقية الأبحاث السريرية أو النظرية. عندنا اللي هي experience must be the work being done in research appropriate to your level of experience and your training. فمهم جدا إنه يكون الشخص خبير في مجال بحثه، وإنه يكون يقدر accuracy before they apply the concepts or procedures. إنه ما يطبق أي إجراء بحثي إلا هو يكون experience في الموضوع أو يكون person will be an expert in the field of research. فمهم جدا إنه يكون best assistance for you choose to think this our procedure for this methodology. عندنا اللي هي السيفتي، نقطة مهمة جدا إنه تعتبر من أخلاقيات البحث، إنه do not expose yourself to the risk of physical or moral, and take precautions when testing the all. مهم جدا أثناء implement for research in environment that may be hazard أو يكون في اللي هي geological أو weather أو social، يكون في أنها ما تتعرض أنها تكون خطرة يعني front the patient. فمهم جدا إنه يكون في concentrate about this. عندنا اللي هو برضو إنه يكون فيه important إنه أنت يعني المستهدفين في البحث إنه ما يكون فيه يعني إحراج لهم أثناء البحث أو يكون فيه تعرض للخطر في موضوع البحث اللي أنت تقدمه. فالسيفتي تعتبر أهم نقطة، حتى لما يكون في برودكت معين أو يكون في بروسيجر لابد أن تقدم على سعودي FDA حتى تطابق المعايير أو البروسيجر before you start your research. ولابد أن يكون في initial monitoring before you start your clinical trial أو procedure with your patient أو your registrations humans with your research. عندنا اللي هي الترست، مهم جدا إنه يكون فيه إن أنت تبني relationship of trust with those who work with them so you get great cooperation and the result with more accuracy. مهم جدا إنه يكون في ثقة بين اللي هو البيشنت وبينك حتى يكون فيه نتائج is excellent with your research. عندنا أهم نقطة اللي هي informed consent. informed consent always make sure you have the اللي هي approve with those who would like to work with them during the period of the research. أيضا إنه مهم جدا إنك أنت تبين للمسجلين في بحثك أو مع المرضى تبين لهم خطوات البحث اللي أنت بتقدمه والبروسيجر اللي أنت بتقدمه للمريض، يقرأ كامل موضح واضح بالخطوات. أيضا إنك تاخذ موافقتهم اللي هو البلانينج and اللي هو الانيشال planning for your procedure أو your treatment when you start with him. عندنا اللي هو لما يكون في withdrawal يعني people have the right to withdraw from the study at any time، إن هم يقدرون إنهم أحقيا إنهم ينسحبون في أي وقت من الدراسة. remember that the participants are all volunteer، يعني هم أول وأخيرا volunteer، أنت بتطبق دراستك وهم متطوعين مشاركين في بحثك، فلهم الأحقية أنهم ينسحبون أو أثناء الدراسة. فيجب أن أنت respect for that and أن يكون فيه برضو reasons you should be expect أنه to the withdrawal. عندنا اللي هي فيه أنه أنت تتوقع أثناء بحثك أو في إجراء اللي هي الدراسة أن يكون فيه انسحاب، فلابد برضو أنه هو أن يكون فيه يعني تأخذ بعد ما يكون فيه withdrawal تأخذ الأبروفة من IRB. وأيضا يكون noted for reason why the patient withdrawal من your research. برضو إن عندنا هنا نقطة أحتاج إنه أنا concentrate with you. في بعض الأحيان إنه الwithdrawal إنه يكون في بداية الدراسة، إنه لسه ما عملت البروسيجر، هذه تعتبر من السكرينينج study، يعني لسه هو ما دخل في الدراسة فما يعتبر هو داخل في الستادي. عندنا اللي هي digital recording، not log photo or take photo or short video without the consent of the target of the search. يعني أنت ما تقدر إنك يعني لابد أن تاخذ الإذن أثناء التصوير أو التقاط صورة من المريض أو المستهدفين من البحث. أيضا ما يمكن إنك أنت تصور وتاخذ فيديو بعدين تستأذن من المريض، لابد أن يكون فيه before you start your take the photo، أن يكون فيه consent approve from your patient أو your registration humans. ولابد أن تكون must be aware that the request for approval after the imaging أن هو unacceptable for your research. عندنا الفيدباك، مهم جدا أن يكون في feedback after your finish for treatment or for your study. If you can give feedback to the target of the search, then you may not be able to provide participants with the report in full, but to give them summary or some phrases and recommendations might be important to have and meet the required purpose. And it's very important to offer them imaging، أن يكون في تصور للغرض اللي أنت اللي قدمته للبيشنت قبل أنت ما تعمل publishing للstudy. So it was not the target of anybody or mental harm because of your interruption or of what they said or did. Make sure you always take pre-profile before publishing. عندنا اللي هي false hope، أنه يكون do not make the target think through questions that think will change because of your search our project conduct by and do not get promise outside، أنه أنت ما تعطي وعود أو خارج نطاق بحثك، أو بمثل أنه إن شاء الله راح أنه أنت تستفيد من هذا الجهاز وراح تستشفي كامل بعد إجراء البروسيجر اللي أنت بتقدمه للبيشنت. فهذا طبعا false hope، أنه من اللي هي ممكن تكون unethical research with your participants. عندنا اللي هي إنه يكون في مراعاة لمشاعر الآخرين، مهم جدا taking into account feeling of others. Some may be more likely to target a feeling of defeatism or surrender because ممكن بسبب إنه يكون الإيج، أو ممكن يكون إنه هو ما يقدر يعرف أو يعني understand أو express your يعني your explain. you should take يعني into account their feeling. فمهم جدا إنه أنت تراعي مشاعر الآخرين في your study، لأنه ممكن يكون هذا unethic لما يكون أنت مثلا not an express for your participant، فهذا يصير unethic of research. عندنا اللي هو exploiting positions، مهم جدا إن أنت ما تستغل المواقف في بحثك، ما تفسر أو بشكل يعني indirect enter the search. مهم جدا إن أنت تكون يعني integrity، ما تكون integrity في your research. فهذه النقطة مهمة جدا. concentrate about your research. عندنا اللي هي السرية، سرية المعلومات تعتبر من الethical. مهم جدا protect identify of the target at all time, not give names or hints lead to the disclosures or identify for another people أو another person. So مهم جدا إنك تحفظ هوية اللي هم البارتيسيبنت، ولا تعطي أي أي كشفية لمعلوماتهم لأي باحثين آخرين بدون ما يكون في consent informed بموافقتهم حتى بعد انتهاء الدراسة. عندنا اللي هي ال principles of ethical research. في principles لابد إن you should concentrate in ethical research. Research should be designed and review undertaken to ensure integrity and equality. مهم جدا إنه يكون في بحثك إنه يكون في نزاهة وإنه يكون في الكواليتي واضحة جدا في your rational questions or procedures in your research. عندنا اللي هو research staff أو subjective must be informed fully about the purpose أو method and intended possible uses of research إنه what participant in research entails and what risk any involve. مهم جدا إنه أنت في بحثك تبين الطريقة والاستخدام الكامل أو البروسيجر قبل أنت ما تبدأ في بحثك، وتبين المخاطر. مهم جدا إذا كانت في مخاطر بحثية في البروسيجر إن أنت تبين للبارتيسيبنت اللي you're applying in your research on them. عندنا برضو السرية زي ما قلنا، هذه البرنسبل اللي ممكن مهم جدا أنت تأخذ بالاحتياط في سرية اللي هي سرية المعلومات. عندنا اللي هي research participant must participant in volunteer away free from any coercion. مهم جدا إن يكون ما في إكراه في الطريقة التطوعية للبارتيسيبنت. لابد أن يكون في respect in your participant. عندنا اللي هي ال harm to research participant must be avoided. اللي هي يجب أنك أنت تتجنب أي ضرر على المشاركين في البحث. وأيضا أنه يكون في independent of research must be clear and conflict of interest، أنه ما يكون في لمصالح أو تحيز يعني واضح في البحث. فمهم جدا أنه أنت you should concentrate about these principles in your research because this and if your صدرت في in your research will be unethical research. عندنا اللي هي النقطة الأخرى اللي راح نتطرقها اللي هي الethical deviations اللي هي الانحرافات الأخلاقية في ethics of research. اللي هي publishing the same paper in two different journals without telling editors. إنك أنت ممنوع إنك تنشر باحث في مجلتين بدون إن أنت ما تعمل أو أن أنت news for editors. عندنا submitting the same paper to different journals without telling the editors برضو ممنوع. مهم جدا أنت تقدم نفس المقالة إلى مجلات مختلفة بدون أنت ما تخبر الإديتورز. هذه تعتبر من المخالفات البحثية. عندنا اللي هي not informing the collaborator of your intent to file a patent. إنك أنت لابد برضو مهم جدا البارتيسيبنت أو التيم اللي معاك الكولابريتر في your research إنه أنت بيكون فيه إنك بتكون إنه أنت patent only inventor يعني في اللي هو your research، لأنه هذه صارت مشاكل كثير وقضايا قانونية يعني شاهدتها وحصلت في الديبارتمنت إنه لما الشخص يحصل على براءة باسمه يعني before أو after procedures، هذه تعتبر من unethical research، فلابد أن تأخذ بعين الاعتبار. عندنا اللي هي including اللي هي colleagues as an author in paper in written for paper even though the colleagues did not make a serious contributions to the paper. مهم جدا أنت تضيف اسم زميلك كمؤلف في البحث. هذه تصير مشاكل كثيرة في أخلاقيات البحث مقابل منفعة لزميل أو يعني conflict أخرى أو إنه not work in your research. عندنا اللي هي discussions with your colleagues، إنه يكون في confidential data from people that you review for a journal. أيضا يكون في terminating outlines from the data without discussions you reason in paper. مهم جدا إنه أنت تحذف النتائج إنه هي تكون سيئة دون مناقشة لتوضيح الأسباب أو limitations. مهم جدا إن أنت you concentrate about the limitations in your research أو determining outlines اللي أنت يعني أنت out in your research. فهذه تعتبر من أخلاقيات البحث. إن صدرت الأوبزيت هي بتصير unethic research، فممكن تصير ignore in some researcher. مهم جدا إن هي تاخذ بعين الاعتبار. عندنا using an appropriate اللي هي التقنية الإحصائية. مهم جدا إن أنت تكون سليمة، إن هدفك إنه يكون في تعزيز rational of your research، ما تكون لأجل يعني غرض معين من غير إنه يكون في benefit for your participant. برضو إنه عندنا البايب سينك بير ريفيو اللي هي تجاوز لجنة مراجعة أخلاقيات البحوث أو إعلان نتائج البحث من خلال اللي هي بريس كونفرنس without يعني ممكن تكون هذه unethic research إنه don't information to review in your work or your team. عندنا اللي هي النقطة الأخرى اللي راح نتطرقها اللي هي conduct review of the lecture، اللي هي knowledge. لابد إن أنت تستعين بمراجع وتشكرها، تقدم شكرها في الأبحاث اللي أنت يعني ساعدوك فيها من أجل إنه out your publishing your research. مهم جدا إنه يكون في knowledge اللي هي acknowledging إنه يكون في شكر للباحثين اللي ساعدوك أو contributions on other people اللي هي peer work. عندنا اللي هي stretching the truth in grant applications، إنه ما يكون في برضو يعني هذه تعتبر من unethic research، إنه أنت تطلب منحة من أجل إقناع اللي هي المراجعات البحثية التي تساهم بإسهام كبير في مجالك. مهم جدا review that your project will make a significant contribution to the field. عندنا اللي هي research publications، نشر البحوث. في لابد إنه إحنا أثناء في ethics steps أو tips لابد إنه إحنا ناخذها بالconcentrate في our research اللي هي honest، إنه يكون في honest and integrity is duty of each author and the expert reviewer and يكون member of journal editor board، إنه يكون في أمانة في الباحث والمراجع وعضو في لجنة أيضا العضو اللي في لجنة النشر. عندنا اللي هي review process contributed to quality control. essential step إنه يكون عملية المراجعة إن هي تسهم في ضوابط الجودة، وأيضا ضرورية في تحقيق اللي هي originality of the research. عندنا الإثيك ستاندرد جورنال إديتوريال اللي هي المعايير الأخلاقية في الإكسبيرينس أوف أن سكروبولوسس أكتيفيتي. فمهم جدا إنه يكون في معايير أخلاقية تقدم بعض الخبرات الأنشطة في المبادئ البحثية. عندنا الأوثرشيب، مهم جدا أثناء التأليف إنه يكون يعني يكون في claim right الأوثرشيب in which order should be author be listed. الauthor must have إنه يكون في contribute to design of study في question اللي هي questions of data analysis and interpretations. مهم جدا إن أنت يكون المؤلف إنه هو يعني شارك في enrollment of data أو في الاناليسيس أو في revision of article أو الفاينل approve أو الversion to publish. فمهم جدا إنه إحنا ما يكون فقط conflict interest في البحث إنه لغرض معين. عندنا اللي هو في التأليف ترتيب كتابة أسماء المشاركين في البحث، إنه يكون في resident، في الديمونستريت، في المدرس المساعد، في اللكتشر، في الأسيستنت بروفيسور والبروفيسور، تكون كذا اللي هي كتابة التأليف. عندنا اللي هي research ethic committee. طبعا هي multi disciplinary. فهي independent and body charged with the review research involve a human participant to ensure that their dignity أو right أو welfare. فهذه هي لجنة أخلاقيات البحث العلمي، هي متخصصة في security أو في protect of a human in the ethic research. عندنا اللي هي research ethics committee must have at least 5 members. في الأخلاقيات البحث هي تكون اللي هي research ethics committee member must have enough experience, expertise and diversity to make informed decision in whether research ethic informed consent sufficient، مهم جدا، and appropriate safeguarded have been put in place. مهم جدا إن هم يكون عندهم يكونوا متميزين في الخبرة، في التدريب، في تنوع العمل، وأيضا يكون في البحث اللي هو making decisions with your research. طبعا عندنا اللي هي work with the studies. الstudies إنه هو عندنا يكون في إنه يكون في نزاهة مع populations. ال REC should have members who are familiar with those group, and ال common for REC include ال advocate أو اللي هم الناس اللي هم السجناء أو اللي هم المعتبرين في new research. عندنا اللي هي النقطة اللي research ethics committee should be include both men and women along with the are not choose specific. لابد إنها تكون تتضمن إنه two groups in your research. عندنا members of research ethics committee must not be all in the same profession. مهم جدا هم يكونوا بنفس المهن مهنة أو الجوبس، لازم يكونوا هم different of the ethics أو الجوبس أو الworks. The research ethics committee must include at least one scientist and at least one non-scientist. مهم جدا إن هم يكونون متنوعين في experience أو في الwork أو في الباك جراوند تبعهم في أثناء اللي هي community of research ethics. ف must not be all of the same professional. طبعا عندنا اللي هي نقطة اللي هي research ethics committee must include at least one person. زي ما ذكرنا إنه ما يكون who is not affiliated with institutions completely called community member. فمهم جدا إنه أخلاقيات البحث على الأقل يكون في عضو واحد تبع المؤسسة البحثية، ودائما يسمى الكوميونيتي ممبر، العضو المجتمعي. الريسيرش ethics committee برضو يجب أن هي تكون فيها consultants مهم جدا discussions to meet requirement for expertise أو اللي هو experience or diversity but only actual REC member. مهم جدا أنهم يكونون مستشارين أو عضو هيئة تدريس من كل تخصص عشان هم يكونون experience أو يكون عندهم باك جراوند أو يكونوا مستشارين في النيور سبيشالست اللي في نيور ستادي. لازم يكون فيه تنوع ويكونون أيضا إن هم يشاركون في الفوت تبع اللي هو نيور ريسيرش. طبعا هذه كانت summary of ethics research اللي يعني كنت أتمنى إنه أنا أتطرق لها، وإن شاء الله كانت واضحة للجميع لأنها point tips more important for your start your research.
شكرا دكتورة مها، يعطيك العافية. يا ريت لو تأكدوا عليا إن الصوت دلوقتي تمام وشايفين السكرين موجودة. أكدوا عليا في الشات لو سمحت عشان نبدأ إن شاء الله. دي آخر محاضرة معانا في اليوم النهاردة. تمام. الكل أكد إن الصوت واضح تمام وشايفين السكرين. في ناس بتطلب 5 دقايق بريك. حاضر تمام، خليهم 8 دقايق إن شاء الله. الساعة 9:10 بالظبط نرجع بإذن الله للشرح. أوكي، بناء على طلبكم. أوكي، 9:10 بالظبط إن شاء الله أنا هستأنف الشرح لآخر محاضرة بإذن الله تعالى. النهاردة هنتكلم عن ال Good Clinical Practice Research Concept، ويمكن دي حاجة مهمة جدا وبتكمل باقي اليوم وبنختمه. دكتورة ما اتكلمت عن جزء الإثكس، وقبلها دكتورة رنين برضو تناولت بعض الأجزاء منها. فاحنا النهاردة هنختم بالكونسبت اللي related بالجود clinical practice research concept. ايه المقصود بيه وليه تم العمل بالكونسبتس دي، أهميتها ايه، تطورت ازاي، نشأت ازاي. وهنقول تطور الموضوع كده وندخل في حتة تاريخية صغيرة جدا عشان نستبين مع بعض ونشوف مدى أهمية فعلا ال guidelines اللي related بالresearch concepts. يمكن امبارح دكتور عبد الرحمن في المحاضرة بتاعته ذكر لكم اللي كان بيحصل وقت الحرب العالمية الثانية، إن German physicians conducted medical experiments على أسرى الحرب أو السجناء without their consent. وللأسف الشديد معظم المشاركين في التجارب دي كان ماتوا بالمئات أو الآلاف، أو حصل لهم عجز أو تشوهات. لذلك كان مهم جدا إن يبتدي يمكن أول حاجة تقريبا على مستوى العالم الحركة اللي حصلت، اللي هي نورنبرج كود 1948، محاولة منهم إن هم يحاولوا يحطوا زي ميثاق أو نقط معينة، إيه بالظبط اللي ممكن يحصل لما نخضع لتجارب، خاصة لو كان human participants. ده كان بداية النشأة للجايد لاينز اللي بنتبعها أثناء الترايلز. آلاف الناس اللي ماتت، آلاف الناس اللي اتشوهت، فكان مهم جدا إن يبقى فيه زي كود. ده كانت البداية بتاعته. طيب هما كان إيه هدفهم؟ أول حاجة قدروا إن هما يحصلوا عليها في الاتفاق ده إن يبقى فيه voluntary consent، إن لازم أي مشارك في التجربة اللي حضرتك بتجريها، سواء كنت عالم في أي مجال أو طبعا المجال الطبي على رأسهم، إن يكون فيه consent. فال-informed consent is absolutely essential. يمكن دي كانت أكتر حاجة خرجوا منها وأهم حاجة من النورنبرج كود. تاني حاجة كان برضو أكدوا عليها إن ال qualified researchers must use appropriate research designs. أكدوا كمان إن لازم يكون في benefit risk ratio عادلة ومقبولة، بحيث إن يكون بقدر الإمكان بنقلل الريسكس اللي ممكن يتعرض ليها المشاركين في التجربة. من أهم كمان النقط اللي اتفقوا عليها في الكود إن participants must be free، من حقهم تماما to stop at any time. طبعا الكل اللي بيسمع دلوقتي يقول لي دي حاجات بديهية جدا، وجود الكونسنت، وجود ان البارتيسيبنت يعترض على التجربة ويوقفها في اي لحظة لوجود ضرر ما بيحصل له. ولكن للأسف هي قبل التاريخ ده 1948 لم تكن شيء بديهي، بالعكس خالص. فكان مهم جدا الكود ده كان هو البداية اللي بدأ بعد كده يتطور مع السنين وفي البلاد المختلفة وفي المقابلات العلمية المختلفة، لغاية ما هيوصل للشكل اللي هنتكلم عليه وهو عنوان المحاضرة ال Good Clinical Practice Research Concept. طيب، وطبعا زي ما اتفقنا حصل بعد كده تطور كبير في هذا السياق وهذا المجال، ومش هنقف طبعا عند كل المحطات التاريخية دي، بس هنذكر أهم واحد منهم وهو ال Declaration of هلسنكي 1964. ويمكن برضو في بعض المحاضرات تناولوه سريعا كده. هو عبارة عن إيه والتطور بتاعه، وفادنا جدا دلوقتي حاليا في إيه ده اللي هنقوله. المؤسسة اللي عملته اسمها World Medical Association. بدأوا إن هم يعملوا development للديكلاريشن ده كethical principles statement، خاصة للميديكال ريسيرش اللي بيتناول human subjects، سواء حتى مش لازم بس بيتعامل مع الهيومن subjects بصورة مباشرة، even حتى لو داتا بتاعتهم ماتيريال خاصة بيهم. وبدأ إن الديكلاريشن ده يتطور وليه كذا فيرجن، ليه 7 فيرجنز زي ما احنا شايفين كده. أول واحد كان في هلسنكي عشان كده الاسم بتاعه، تاني واحد كان في طوكيو، التالت كان أو التاني كان في فينيس، التالت في هونج كونج، وهكذا. كل سنة كان بيبتدي إن هم يجتمعوا مع بعض ويطوروا أكتر في الديكلاريشن ده، ويحطوا رولز أقوى، يبتدوا إن هم يعملوا development للانترناشونال جايد لاينز بشكل أقوى. وطبعا كان في مشاركات من جميع البلاد بشكل انترناشونال، ولكن مع التطور ومع وجود كل سنة طبعا بيطلع، وجدوا إن في أوقات تضاد ما بين الارتكلز اللي بتطلع كل سنة، وإن كمان أصبح في جزء منها أكثر تعقيدا شوية. فبدأ الاعتماد على ال DOH اللي هو ال Declaration of Helsinki يتراجع شوية، بالرغم إن الدوكيمنت ده very comprehensive، كبير إن ال subjects يتم حمايتهم في مختلف الدراسات، وكان مهم جدا is very essential for protecting the patients. ولكن أكيد أي شيء لا يخلو من الخطأ، ولذلك ما بقاش يتم تداوله أكثر، لأن زي ما اتفقنا كان في بعض contradictory وجود تضاد أو unclear articles فيه. فكان هو ده الانترودكشن الحقيقية للجود كلينيكال براكتس، ويمكن دي من أهم فوائده، إن بدأنا ناخد منه جزء ونعتمد أكتر على تطوير وصياغة الجود كلينيكال براكتس جايد لاينز اللي هنتكلم عليها. إيه هي الجود كلينيكال براكتس اللي هنتكلم عليها؟ الجود كلينيكال براكتس اللي اختصار GCB is International Ethics Scientific and Practical Standard by which all clinical research is conducted. هي عبارة عن International Ethics Scientific and Practical Standard اللي عن طريقها أو من خلال خطواتها أو محاذيرها الكلينيكال ريسيرش بيحصل له إنه يترجم على أرض الواقع. everyone كل شخص باختلاف موقعه، سواء كان من أول الشخص الانفستيجيتور أو المسؤول عن البحث أو المنفذ للخطوات IRB زي ما هنتكلم، كل شخص involved في الريسيرش بروسيس must، مهم جدا إن هو يكون trained وفاهم جدا يعني إيه good clinical practice guideline. يعني الموضوع ما بقاش اختيار زي الأول، وإنما بقت كل حاجة خاضعة لمعايير دولية ومعايير معترف بيها. وطبعا التطور العلمي الرهيب اللي احنا الحمد لله بنعيشه دلوقتي بيفرض إن كل المؤسسات البحثية، كل شركات الأدوية، كل الجامعات الكبيرة لما بيكون مطبق فيها ترايل، خاصة الترايلز الكبيرة اللي بتتعامل تعامل مباشر مع الهيومن سبجكتس، بيكونوا حريصين جدا إن يتم تطبيق كل المعايير for good clinical practice. طيب ممكن نسأل سؤال ونقول why ال good clinical practice for research is so important؟ ليه التريننج عليه مهم؟ زي ما اتفقنا for all staff members involved in clinical research ال training for basics بالذات good clinical practice is very important. هو أمر غير خاضع للنقاش أو المفاوضة. مهم جدا إن أي research باختلاف ال study designs طالما involved with human subject must adhere to good clinical practice guidelines. نقطة كمان برضو مهمة جدا إن ال good clinical research practice هو its general concept can be applied to any research protocol. ممكن حضرتك تطبقه باختلاف ال research designs، باختلاف ال research protocols، طالما بتتعامل مع human subjects بشكل مباشر. It's not protocol specific. ملهوش علاقة معينة بنوع دراسة أو بروتوكول معين أنت بتتبعه أو approach معين، وإنما طالما بتتعامل مع human subject فواجب عليك أنت والتيم من أوله لآخره تكونوا بتطبقوا ال good clinical research guidelines. يبقى اتفقنا إن ال good clinical research guidelines أو practice are very important, very essential، وشيء حاسم جدا required for everyone who will be directly بشكل مباشر هيتعامل جوه research project involving human participants. What are the goals أو إيه هي ال goals for ال GCB؟ أول حاجة to protect the rights، protect the safety and welfare for human participants، حماية الحقوق ورعاية الأشخاص الخاضعين للتجارب، حمايتهم من التعرض لأي أذى محتمل لإجراء البحث مثلا. دي كانت من أهم الجولز فور الجود كلينيكال براكتس، عشان كده أنا بأكد جدا على أهميته. رقم 2 to assure the quality and reliability and integrity of collected data، التأكيد والمحافظة على الكواليتي الجودة والريلابيليتي والانتجريتي الصحة والسلامة وتنسيق الداتا اللي حضرتك بتعمل لها collection من الهيومن subjects اللي بتتعامل معاهم. رقم 3 to provide standards and guidelines to conduct clinical research. لذلك نقدر نلخص إن ال good clinical practice equal applying ethics زائد assuring quality of data زي ما هنشوف مع بعض. لو هنلخص ال good clinical practice إيه هو applying ethics plus assuring quality of data. So let's say إن ال good clinical practice is scientific quality standard for designing رقم 1 for conducting, recording and reporting human subject clinical trials. بنستخدم المعايير بتاعته من أول ال designing، ال conducting إجراء التجربة، recording تسجيل البيانات، التعامل مع كل ال human subjects اللي موجودين في التجربة، وكمان في ال reporting الداتا اللي طلعت لينا. When the good clinical practice guidelines were proposed in collaboration with International Council for Harmonization، وده اللي هنتكلم عليه. وممكن تلاقوا وانتوا بتقروا مثلا الاختصار ده ICH-GCB، ده اختصار للجايدلاينز اللي هي كان توافق ما بين الجهتين أو التو كونسبتس عشان نطلع أفضل جايدلاينز ممكنة نقدر نستخدمها. واتفقنا إن الهدف هو to protect rights, safety, privacy of clinical trial participants. ده الهدف الأول والأسمى والأهم، لأن مش منطقي أبدا إن حضرتك تدخل معاك participants بهدف أيا ما كان هدفك العلمي سامي ونبيل، بس تسبب individuals. ويمكن أنا اشتغلت مع الأشخاص دول عن قرب جدا مع السباينال كورد انجري individuals. بيبقى ليهم طبيعة طبعا خاصة، بيبقى ليهم طريقة معينة مناسبة للتواصل معاهم. even حتى لو بتاخد منه كويشنير وبتسأله أسئلة، مهم جدا إن أنت يكون عندك ما يكفي من الكياسة والفطنة وكلامك دمس إن أنت ما تسببلوش زعل أو ضيق معين، وإن هو أكيد من حقه في أي لحظة يرفض يكمل الدراسة. وده بيبقى معظم الدخول بيبقى مدفوع الأجر، بس يكفي إن هو طبعا أكيد إنسان عنده ظروف خاصة، فمهم جدا إن نضمن إن مفيش أي إصابة نفسية أو معنوية أو اجتماعية أو طبعا جسدية أي بارتيسيبنت هيكون معاك بيتعرض لها بأي شكل من الأشكال تحت أي اسم من الأسماء وتحت أي ظرف من الظروف. ده مهم جدا يعني. تاني من الأهداف برضو المهمة to ensure إن ال data collected in clinical trials بالإضافة طبعا للنتائج اللي طلعت بناء على هذه الداتا هي accurate and credible، لأن طبعا كل تجربة أو كل قرار conclusion و results خارجة من التجارب، بالذات التجارب الكبيرة اللي بيتم تطبيقها والاعتماد عليها جوه الكلينيكال براكتس بتكون حاسمة جدا وبتكون مهمة جدا. فالجايد لاينز دي مش بس بتهتم بالبرايفسي والسيفتي والرايتس للبارتيسيبنت، وإنما كمان بتكون مهمة لأنها بتعمل بشكل أو بآخر ضبط لعملية الداتا كولكشن، الكواليتي أشورنس للريزلتس اللي هتطلع، إنها تكون accurate بقدر الإمكان وكريديبل. الاختصار اللي كنت بقول لكم عليه ICH GCB اللي هو International Conference on Harmonization Good Clinical Practice، ده الرمز بتاعهم. الجايد لاينز دي عبارة عن تكاتف الكارنت جود كلينيكال براكتس في الاتحاد الأوروبي، في اليابان، في اليونايتد ستيتس، كمان في أستراليا وفي كندا والنورديك كانتريز والورلد هيلث أورجانيزيشن. فهو التكاتف بتاع الجايد لاينز في البلاد اللي احنا ذكرناها دي بيطلع مفهوم الجود كلينيكال براكتس. طيب ايه هي البرنسبلز يا ترى؟ يعني ايه هي البرنسبلز الأساسية اللي مستند عليها الجايد لاينز؟ هنتكلم عن الإثكس، هنتكلم عن الساينس اند بروتوكول، هنتكلم من أهم البرنسبلز responsibilities، عن الكواليتي أشورنس أو الكواليتي كنترول، هنتكلم عن الداتا انتجريتي اند كواليتي، هنتكلم عن الكونسنت with knowledge. ويمكن زي ما قلنا في المحاضرات اللي فاتت مع دكتورة رنيم أو دكتورة مها أو دكتورة عبد الرحمن تطرقوا لبعض النقط دي بالتفصيل، زي دكتورة مها اتكلمت عن الإثكس، أعتقد دكتورة رنيم اتكلمت عن الكونسنت، دكتور عبد الرحمن اتكلم عن الريسبونسابيلتيز. فمهم جدا نفهم إن دي الركائز الأساسية اللي بيتبني عليها ال 13 principle for GCB: الethics, science and protocol, responsibilities, quality assurance أو quality control, data integrity, consent. دي الركائز الأساسية لل 13 principle. هنمسك واحد واحد فيهم ببساطة ونشرحه بسهولة جدا إن شاء الله. أول حاجة المفهوم for ethics. Clinical trial should be conducted in accordance with the ethical principles. ده شيء لا يوجد فيه أي نقاش أو جدال، أكيد متفقين جميعا عليه إن we should follow the ethics. وعشان كده أنا بدأت المحاضرة بالتعريف إيه هو Declaration of Helsinki، لأن الجزء ده متاخد منه. رقم 2 من ضمن ال principles trial risk versus trial benefit، ويمكن دي كانت من أهم الحاجات اللي بنقابلها في لما حد عنده بقى يعني حماس كده وعايز يقرأ أكتر عن التجارب البشعة المميتة اللي اتعملت في الحرب العالمية التانية. للأسف الشديد ما كانش فيه أبدا الكونسبت ده، كان ضايع تماما، اللي هو risk versus trial benefit. قبل الترايل، قبل الكلينيكال ترايل ما يتم بدأها والشروع فيها، مهم جدا إن القائمين عليها يعملوا ميزان بمنتهى العدل والشفافية والاحترافية، إن هم يعملوا موازنة ما بين الريسك وما بين البنفيت، خاصة طبعا بالنسبة مش بس المجتمع العلمي أو ليهم، وإنما كمان مهم جدا إن احنا to take care from subjects. Okay. So trial should be initiated and continued ونستمر فيها فقط if لو ال benefits justified the risks. لو في الريسكس اللي هتحصل حضرتك قادر ان انت تعمل لها كنترول وتسيطر عليها، ما تبقاش مدمرة او مميتة، وفي نفس الوقت البنفيتس كفة الميزان فيها اعلى واقوى، في الحالة دي you can make the trial. unless that you can't. البرنسبل رقم 3 trial participants: the right and safety and well-being of the trial subjects are the most important considerations. الحقوق والحماية وصحتهم إنها هتكون جيدة، مش هيحصل لهم ضرر من الترايل، هو أهم concept أو أهم consideration لابد الأخذ فيه بعين الاعتبار. يمكن كمان بيغلب على مصلحة المجتمع أو even الهدف العلمي من إجراء التجربة، هو prevail over the interest of science and society. وبالتالي مهم جدا إن بيكون في، طيب مين اللي بيقيم ده؟ طبعا ال IRB، ال Institutional Review Board، اللي بتتم مراجعة البروتوكول زي ما هنتكلم مع بعض، إنها بتحاول بقدر الإمكان تقرر وتقيم مدى البنفيتس ومدى الريسكس اللي ممكن يتعرض ليها السبجكتس جوه الترايل. البرنسبل اللي بعد كده information on the medical product. مهم جدا إن ال available non-clinical and clinical information for the product should be adequate to support the proposed clinical trial. مهم جدا إن ال clinical information and non-clinical information تكون كافية وواضحة للبرودكت أو الدراج أو أي مكان اللي بيتم البحث فيه. كل clinical trials should be scientifically sound and described in a clear detailed protocol. ويمكن احنا في المحاضرة بتاعتي معاكم اتكلمنا شوية عن البروتوكول وحابة أراجع عنه. يمكن من أهم الملحوظات أو المعلومات اللي تخرجوا بيها من الكورس البروتوكول، لأن احنا زي ما اتفقنا البروتوكول بس مش حاجة خاصة ممكن بالباحثين، وإنما أنت ممكن تكتب بروتوكول في أي حاجة ممكن تعملها. وإن كان البروتوكول للكلينيكال ترايلز أو للكلينيكال ريسيرش طبعا أكيد مختلف، بس يبقى إن قواعد الكتابة العلمية اللي اتفقنا عليها امبارح وأول هي ثابتة طبعا. اتفقنا قلنا إن في فرق ما بين البروتوكول والبروبوزل. البروتوكول هو عبارة عن plan for research study، وكذلك ده تعريف البروبوزل plan for research study، ولكن في فرق في أهمية كل منهم. كل واحد فيهم ليه هدف، وبالتالي بيختلف شوية عن الجزء اللي انت بتركز فيه وتبرزه وتظهره. اتفقنا إن البروتوكول هو عبارة عن تقديم المنهجية البحثية بالتفصيل، عشان حضرتك بتقدمها for ethical consideration. مهم جدا بتكون ذاكر البارتيسيبنتس، هتسجلهم ازاي، هتتعامل معهم ازاي، هل في randomization تم ازاي، الالوكيشن concealment للبارتيسيبنتس، وليكن بتجرب دراج مثلا او دراج A versus B، او بتشوف السيفتي والافكسي اوف any technique. فمهم جدا بتذكر كل شيء المنهجية البحثية بالتفصيل، حجم العينة بالظبط، احتسابها، طريقة احتسابها، الدوز، السيفتي، الفولو اب، الاسسمنت، كل ده بيتم ذكره بالتفصيل جوه البروتوكول. بينما البروبوزل هو برضو still plan for research study، ولكن أهم حاجة بيتم التركيز عليها التايم لاين، البادجت، الاكسبكتد outcomes، لأن هدفه الأساسي not ethical consideration زي البروتوكول العادي، وإنما هو بيكون عبارة عن طرح مادي للوقت المحدد بالظبط لكل خطوة، الوقت وكمان الفلوس اللي أنا محتاجاها أو النفقات الميزانية، لأنه عادة البروبوزل ده بيقدم للجهة المانحة للحصول على جرانت، سواء شركة أدوية، شركة أجهزة، أيا ما كان الجهة المانحة الممولة للبحث. اتكلمنا برضو امبارح وقلنا البروتوكول بيتكون من إيه؟ قلنا فيه أجزاء ثابتة، وإنما بتفرق من جامعة للتانية، من مؤسسة بحثية للتانية. لما حضرتك بتكون بتقدم على بروتوكول مهم جدا إن أنت تتواصل معاهم وتسألهم هل في تمبلت معين أنا بمشي عليه. ومعظم الجامعات institutional places عندها بروتوكول تمبلت انت بس بتملاه. طيب ايه اهم الستراكشرز اللي في اي بروتوكول عامة في اي مكان غالبا بتبقى محتاج تكتب الاشياء دي؟ قلنا اول حاجة introduction اللي بتتكون من جزئين background and stating the problem and rationale. تاني جزء aim and objectives. aim it is the main goal but objectives هو minor goals. رقم 3 مهم جدا ولسه قايلين methods. You should describe the study design, the place, the population, the sample size, the data collection, the data analysis. كل الإجراءات الميثودز اللي انت عملتها لابد انها بيتم او ان انت هتعملها طبعا لان ده بروتوكول في صيغة المستقبل بيتم ذكرها بالتفصيل. الجزء الرابع بيكون مهم جدا في البروتوكول وهو الإثكس. بتبتدي ان انت وده بقى شغلنا النهارده بتبتدي تذكر ايه هي البنفيتس اللي ممكن يكتسبها العيان من دخوله في التجربة دي. بتبتدي تكتب الهارمز اللي ممكن تحصل له، المشاكل اللي متوقعة إنها تجراله، وهل يا ترى لو حصل أي مشكلة ما في الطريق وأنت ماشي بتكون أو أي سايد إفكت ظهر هتبتدي تتعامل معاه إزاي. وطبعا بيكون في جزء الانفورمد كونسنت مذكور جوه البروتوكول وهيتم التعامل ازاي مع العيان وازاي بتاخد موافقته، وهل مثلا لو العيان أمي لا يقرأ ولا يكتب هتتعامل معاه ازاي مثلا، هل بتاخد منه مثلا موافقة شفهية في حضور شهود، دي طريقة من الطرق. طيب لو العيان ده مثلا كان وليكن كفيف مثلا أو أيا ما كان على حسب العينة البحثية بتاعتك بتبتدي إن أنت تذكر جزء الانفورمد كونسنت بتاخد وقت، وليكن لإما الموافقة الكتابية بمنتهى الاستنارة بعد ما بتشرح للسبجكتس بتوعك إجراءات التجربة بالظبط مدى البنفيتس اللي ممكن تعود عليهم أو الهارمز اللي ممكن يتعرضوا لها، تشرح لهم البروتوكول كامل، الفريم اوف تايم، كل شيء بيتم شرحه ليهم وكل ده بيتم ذكره جوه الكونسنت المفروض، بهدف ان اتفقنا عشان اقدم الاي ار بي اللي هي institutional review board عشان اخد الموافقة. وكل ده من ضمن الجود كلينيكال براكتس جايد لاينز اللي بنتكلم عليها. البرنسبل اللي بعد كده قلنا عليه good quality trials، ان الكلينيكال trials should be scientifically sound، مهمة جدا وزي ما اتفقنا موصوفة بديتيلد بروتوكول واخدين عليه الموافقة مسبقة. البرنسبل اللي بعد كده بيأكد على نفس الجملة لأن البروتوكول هو most important document in the study، ما فيهاش نقاش. الجزء ده مهم جدا وانه مهم زي ما اتفقنا يكون بيحتوي على الستادي ديزاين بالتفصيل، البيربز راشونال، الانديكيشنز، الاند بوينت، الايفالويشن والاسسمنت، كل ده should be clearly described in the protocol. يمكن هنا حابة أقول لكم سريعا الفيزز of clinical trial عشان نتكلم باستفاضة شوية صغيرة على الفيز الأخيرة. أول فيز no control group. هما الفيزز of clinical trials عادة بتبقى 4 phases. أول حاجة no control group just small size sample ممكن من 10 ل 20 participant just to investigate the safety، بنشوف السيفتي للدراج ده أو للتكنيك بس فقط لا غير. طبعا في control group السامبل سايز ممكن يزيد حاجة بسيطة وليكن من 100 ل 200 subject. هنا الهدف our aim is to prove the concept and explore efficacy. الفيز رقم 3 for clinical trials في control group أكيد، السامبل سايز بتكبر وممكن في بعض الترايلز الكبيرة القوية اللي التكلفة بتاعتها عالية توصل للآلاف، وهنا الإيم بتاعنا to test efficacy and control السايد effects. طيب جميل، أنا عملت خلاص، ليه بقى بحتاج الفيز رقم 4؟ الفيز رقم 4 مهمة جدا ولا تقل أهمية عن اللي فات لأن هي بين قوسين بنسميها post marketing evaluation. post marketing evaluation بنحاول إن احنا نشوف الأعراض الجانبية الممكن حدوثها واللي ما ظهرتش في أي فيز من اللي فاتت خالص، لأن احنا وليكن أنا يعني ذكرت ده امبارح في الشرح، قلت لكم إن عالم التجارب السريرية، عالم الكلينيكال ترايلز، احنا بنختار فيه typical patients أو typical subjects. عادة بيتم اختيار الناس بدقة شديدة وبعناية شديدة. غالبا مثلا بيكون مش عندهم comorbidity سابقة، بيكون في غاية الالتزام إن هم يتناولوا الدراج أو التكنيك اللي احنا بنقيسه. حضرتك بتكون مهتم بيهم جدا أثناء إجراء التجربة، كل شيء بميعاد، كل شيء مظبوط، في فولو أب. فبالتالي لما بنعمل بوست ماركتنج ايفالويشن بيكون النتائج أصدق وأعم وأشمل، لأن هي بتتجرب على السوق بشكل عام، على الناس أو المرضى أو الجمهور اللي ممكن يكون فيه atypical subjects كتير جدا، اللي بيكون فيه اختلاف الناس، اختلاف الأعمار، اختلاف الأجناس مثلا، أو العرقيات. طبعا ده ممكن يفرق في النتائج وليكن. وممكن كمان بيكون في بعض سايد افكتس هي من طبيعتها انها ما بتظهرش الا بعد وقت طويل. فالبوست ماركتنج ايفالويشن ده بيبقى فيه غاية الأهمية. يمكن دي حتة كده حبيت ان انا اتطرق لها في النص، الشيء بالشيء يذكر يعني، لانها حتة مهمة. اتكلمنا عن المين كومبوننتس اوف ستادي بروتوكول، قلنا الانترودكشن اللي بيكون فيها الباك جراوند والراشونال طبعا والبروبلم والايمز اند اوبجكتيفز والميثودز، وقلنا الميثودز بيتم ذكر فيها ايه وليه، وبتكون دي من أهم الأجزاء اللي بيتم تقييمها، هي والethical considerations من ال IRB. وطبعا ال informed consent فيه كل التفاصيل اللي محتاج المريض يعرفها، وفي نفس الوقت الموافقة بتاعته. ومهم جدا برضو نعرف إيش هي الراشونال. في حد بيسأل سؤال جميل، برافو عليكي. تمام، هقولك حاضر. الراشونال هي الجذر العلمي للمشكلة، ايه هو الأساس العلمي للموضوع. وليكن مثلا أنا قلت هستخدم الليزر لعلاج الكاربال تانل سيندروم، المثال اللي اتكلمنا فيه امبارح. ايه هو الراشونال بتاع استخدام الليزر؟ هل مثلا ان الليزر انا بعتبره انتي انفلاماتوري، وان هو مهم لكذا كذا كذا في الكاربال تانل سيندروم لان هي من ضمن خصائصها كذا، فاستخدام الليزر في الخصائص دي هيقلل من النتيجة؟ يبقى الراشونال هي الجذر العلمي للموضوع، للمشكلة نفسه، ايه علاقة الحاجة ببعض من الناحية العلمية. فطبعا ده بيتكتب في الانترودكشن، بيتم ذكره لربط الفاريبلز ببعض. اوكي، الاوبجكتيفز بقى مهم قوي ناخد بالنا ان الاوبجكتيفز فور بروتوكول لازم او مهم يكون فيها كلمة سمارت متحققة، S for specific، M for measurable، A for attainable، R for realistic، T for time bound. مهم جدا ان انا مش بكتب objects كده في الفضاء الخارجي او بدون هدف، وانما لابد انها يكون متوفر فيها كل الكرايتيريا دي. اوكي، احنا ماشيين في باقي البرنسبلز للجي سي بي. البرنسبل اللي بعد كده الميديكال decisions. The medical care given to الميديكال كير and medical decision للسبجكتس اللي خاضعين للتجربة مهم جدا إنها تكون مسؤولية qualified physician. وده شيء في غاية الأهمية، لأن أولا خلال التجارب باختلاف طبعا درجة التجربة أو كبرها أو حجمها ممكن نتعرض لوجود side effects، نتعرض لوجود مشاكل ما. فأي قرار يتاخد أو even حتى الويجوال كير مهم جدا إن هو يكون مسؤول عنه qualified physician. ولو كان حاجة related مثلا بمجال الأسنان أكيد هيبقى qualified dentist، وهكذا على حسب المجال اللي بيتم تطبيق فيه الترايل يعني. البرنسبل اللي بعد كده مكمل للبرنسبل اللي قبله، بيتكلم عن الستاف بشكل عام، مش بس الجزء اللي خاص بالبروفيشنال أو الكواليفايد physicians اللي هما بيطبقوا التجربة، وإنما الستاف بشكل عام. Each individual involved in conducting a trial should be qualified، مهم جدا by education training and experience للتاسك اللي مسؤول عليها أيا ما كان موقعه إيه جوه الترايل. من أول وليكن ال data collection، ال dealing with subjects، ليه جزء في ال intervention، بيعمل evaluation و assessment، بيتولى ال data entry وال data analysis. طبعا كل الأجزاء دي مهمة جدا، لأن أي خلل في أي مرحلة من المراحل دي بيأثر جدا على النتائج. وإحنا اتفقنا إن من أهم الأهداف للجود كلينيكال براكتس هو إن النتائج تكون verified ويمكننا الاعتماد عليها. البرنسبل اللي بعد كده هنتكلم عن ال informed consent. Freely given informed consent should be obtained from every subject. طبعا أكيد قبل الترايل participation، قبل المشاركة جوه الترايل، مهم جدا إن كل subjects حتى لو كان عددهم بالآلاف أيا ما كان العدد، كل واحد قائم بذاته بيدي ال informed consent بناء على معلومات في غاية الشفافية والمصادقة والمصارحة اللي مجرين البحث بيتناقشوا معاه فيها. النقطة اللي بعد كده ال clinical trial data. all clinical trial information should be recorded. أكيد وألف ب بحث علمي إن كل حاجة تبقى منظمة ومسجلة وقابلة للرجوع فيها في أي وقت. فمهم جدا التعامل مع الداتا for subjects أو patients بشكل خاص، إنها تكون recorded well، can be handled and stored in way بطريقة very accurate وقابلة للانتربريتيشن بسهولة and very verified، ونقدر نرجع لها في أي وقت بسهولة. النقطة اللي بعد كده ال confidentiality أو ال privacy. إزاي نقدر إن احنا نحافظ على ال privacy respecting ال privacy for each subject. دي من أهم النقط ك requirement لل guidelines for good clinical practice. النقطة اللي بعد كده good manufacturing practice. البرودكتس اللي بيتم البحث فيها أو تناولها جوه الترايل should be manufactured, handled and stored in accordance with applicable good manufacturing practice. وده أمر مهم جدا، لأن احنا هدفنا حماية المستهلك أو حماية السبجكتس جوه الترايل. فبالتالي المواد اللي بيتم استخدامها أو الدراج اللي بيتم التجربة عليه أو أيا مكان ايه اللي بيتم تجربته، احنا بنتكلم طبعا بشكل عام بس معظم الترايلز طبعا تبقى فور دراجز يعني. مهم جدا انه يكون خاضع للجود مانيفاكتشرنج براكتس جايد لاينز برضو الخاصة بالمادة اللي بيتم الشغل عليها، ويكون ده متوافق وماشي مع الابروفد بروتوكول، لان زي ما اتفقنا ان سلامة الشخص اللي انت بتجري عليه التجربة من اهم الاولويات. البرنسبل اللي بعد كده ال quality assurance. systems with procedures that assure the quality of every aspect of trial should be implemented according to quality assurance. كل دول لو لاحظنا معظم البرنسبلز ال 13 بيدور في نطاق تحقيق البيشنت سيفتي، ولأنها من أهم المتطلبات والعوامل اللي على أساسها تم عمل وتطوير الجايد لاينز اللي بنتكلم فيها. البيشنت سيفتي دي عبارة عن إيه؟ عبارة عن تأكد سلامة وصحة الشخص المشارك في التجربة من كل الجهات الممكنة. Ensuring participant safety include among other things من أول designing protocol، monitoring، quality assurance، adhering to rules، ethical consideration. كل مراحل الترايل هي بتكون مركزة جدا وهدفها الأساسي ضمان وتحقيق البيشنت سيفتي. يمكن كلمة سيفتي كلمة جميلة قوي، هي مش جاية من فراغ وإنما كل حرف فيها اختصار لمعنى. S for sense the error، A for act to prevent it، مجرد الاستشعار إن في مشكلة أو خطر ما على البارتيسيبنت، محاولتك ان انت تمنعه قبل حدوثه، مش بس معالجته لو حصل، انت دورك كمان ان انت كمشارك في الترايل. وطبعا جزء كبير دي من دور ال IRB قبل ما تدي الموافقة أصلا لإجراء التجربة، هم بيحاولوا بقدر الإمكان إن البيئة تكون safe to prevent أي error ممكن يحصل. F follow safety guidelines، في guidelines بنتبعها. E for inquire into accident أو death. T for take appropriate remedial measure. why your responsibility، ويمكن دي ال responsibility من ضمن، لو نفتكر فوق، هي دي من ضمن الركائز الأساسية اللي بيتم بناء ال good clinical practice عليها، إن كل شخص جوه الترايل عليه responsibility. من الأول ال investigators، ال IRB، مجرين البحث والمطبقين، even حتى ال subjects ممكن يكون عليهم جزء من المسؤولية. مهم إنهم يكونوا فاهمين كل الخطوات وطبعا موافقين على إجراء التجربة. فخلينا نقول سريعا أهم الأهداف اللي على أساسها بيتم أهمية أو تلخيص أهمية فعلا الجود كلينيكال براكتس. زي ما اتفقنا، هو البيشنت سيفتي، هو نتأكد إن البيشنتس will be safe، أو بشكل عام مش شرط بيشنتس بس، أي مشارك جوه التجربة. وإن الجود كلينيكال براكتس هي عبارة عن applying ethics. أنا قلت لكم الجملة دي في أول المحاضرة، قلت لكم هتفهموها أكتر في الآخر، إن الجود كلينيكال براكتس هو عبارة عن applying ethics زي ما شرحنا، زائد assuring quality of data. لو طبقنا المعيارين دول أو تأكدنا إن الاتنين موجودين، بالتالي كنتيجة متوقعة هيبقى فيه safety للبيشنتس، هيبقى فيه adequate results يمكننا الاعتماد عليها، هيبقى فيه كل الإجراءات السلامة والأمن اللي اتكلمنا عليها of data. احنا كده الحمد لله خلصنا المحاضرة النهاردة، وشكرا جدا على استماعكم. لو حد عنده أي سؤال أو أي استفسار أو حاجة ممكن يكتبه. عفوا.